Okay, maybe we get started. That's okay. So my name is Devin Krogan. I'm the director of the Quantitative Biosciences Institute, and it's my pleasure to welcome you to the latest installment of the QBI Symposia series entitled Frontiers in Epigenetics and Chromatin from Fundamentals to the Clinic, organized by Denitsa Fujimori and Gita Narlikar. And we have a very exciting lineup that they've put together, everything to do with chromatin, chromatin function, chromatin biology, chromatin structure. So if you are excited about chromatin, this is the place to be over the next couple of days. And I just wanted to highlight the fact that this is a symposium that's in a long line of QBI symposia that we've been having over the last several years. Before the pandemic, obviously, we were having these in person, both here and abroad. Obviously, more recently, we've been having these virtually. And I just wanted to highlight one that's coming up next, just very briefly. This is one that's led by the QBI fellow, Willow Coyote Mestis. I think it's a unique one because it's focused on scientists, native and indigenous scientists exclusively, that are working in quantitative biology. And it's November 18th and 19th. So I encourage you to put that on your calendar. And I just wanted to thank the individuals that help on all of these symposia, both of these and the ones that we've been having over the last several years. This is the QBI team that's been working closely with Gita and Denitsa, Jacqueline Fabius, Alexa Record, Gina Wynn, Karolina Vreni Lenzi. So I'd like to thank these individuals. They often don't get the credit that they deserve. And the fact that everything runs so smoothly is a testament to the quality of their work. And with that, I will just hand this over to two of the best here, Denitsa Fujimori and Gita Narlikar.
Thank you, Nevin. And I just first want to welcome everybody. Welcome to this symposium on chromatin and epigenetics. We hope you enjoy the next few days. We have assembled a set of speakers who you will see are truly doing frontier research in chromatin and epigenetics. So you're in for a treat. And without further ado, I am going to introduce our first speaker, Eva Nogales from Berkeley, where the time is currently 8:33 a.m. I'm going to do this for every speaker, so you appreciate that they're coming from different parts of the world. Eva, all yours.
Thank you, Gita and Denitsa, for putting together this and for inviting me to present. I'm not sure I'm so thankful about going first, but that's the way it is. So today I'm going to be telling you about what we found out concerning the modularity in the structure and functionality of some histone-modifying complexes. In particular, I'm going to be telling you about histone acetyl transferases. So SAGA is a prototype of this modular set of modular complex with different sets of functionalities. It shares a core and a structural core with a complex that we love and we studied a lot, which is TF2D. But it also shares its enzymatic modules, the HAT and the D-ubiquitinates with a number of other complexes. And the trap subunit that is involved in recruitment by interaction with sequence-specific activators is shared with another histone acetyl transferase. In this case, instead of H3 like SAGA, it modifies H4 and H2A lysins, and that's the NUA4. So in the case of human SAGA, the modularity and the sharing continues because human SAGA contains subunits shared with the HSF3B splicing module of the U2 SNRNP. And there are other subunits that are actually, there's a subunit that is specific to yeast, which is the SPT8, which in yeast is involved in interaction with TBP. This subunit is not present in metazoans, is not present in human, which is the one we studied. And in that particular case, we don't know whether TBP is actually engaged by SAGA. In any case, I'll be telling you very briefly about TF2D, just concentrating on the modules that are shared or are homologous to the ones present in SAGA. Then I'll tell you about SAGA and end up telling you about our recent unpublished studies on the yeast NUA4 complex. And you will see that there are shared modules that may or may not have the same functionality. They may diverge in what it is that they do in the complex. So TF2D is a large transcription initiation complex that is involved in recognizing Pol2 core promoter sequences and then assemble the pre-initiation complex. Is made up of 3 major lobes, 2 of which are related, lobe B and lobe A. So lobe B contains 3 pairs of TPP-associated factors or TAFs that contain histone folds, and they pair TAP9 with 6, 4 with 12, and 8 with 10. And they bind another subunit, which is the TAP5, containing WD40 beta propeller domain and an all alpha helical N-terminal domain. This is repeated in lobe A, except that TAP8 is substituted by TAP3. And now this forms a module that can recruit a 4th pair of histone fold containing subunits, TAP11 and 13, which themselves bind and engage TBP. So these 2 related modules with a common scaffold and a slight difference play completely different roles within TF2D. Lobe A, which is depicted in yellow with TBP in red, undergoes very large conformational changes. Its role is to keep TBP inhibited until it is the right time and position to deploy it. Lobe B, which interacts more rigidly with the rest of the complex, contribute to defining a DNA molecular ruler within TF2D, and then helps deploying TBP by interaction with TF2A. The final result of the combined action of these 2 modules with the module that bind the downstream core promoter elements is the formation of this structure that is now deployed TBP and can recruit the rest of the pre-IC and bring the polymerase to the transcription start site. So as I said, that histone fold containing a scaffold of TAFs is present also in the Saga histone acetylase complex. And in this case, it serves really as a structural hub for interaction with the rest of the functionalities in the complex, which are the HAT and the DAB, as well as interaction with the large trap subunit that interacts with sequence-specific activators. So the structure of Saga for 2 yeast species was reported about a year and a half ago from the labs of Patrick Kramer and Patrick Schulz, and they were able to get reasonable resolutions and build models for the TRA1 and the core element, while the HAT and DAPs were actually very flexible, something that is you will see as a very common theme. The human has a number of differences. The sequence is not very well conserved. There are insertions, deletions. SPT8, as I said, doesn't exist in human, but there is an splicing module. And while Saga is not essential in this, it is required for metazoa and embryonic development. So we decided to pursue the human complex and make and purify it from endogenous sources using a tag, CRISPR engineered tag in one of those subunits that is unique to Saga. And then Dominic in my lab obtained the structure. As before, there are rigid parts, so the trap subunit and the core are well resolved, are better than 3 angstrom. He could model directly into that. For domain of the TAP6-like subunit and the splicing module that attached to it, that's flexible. We could obtain it at lower resolution, but thankfully we could interpret it based on both homology model for the hit repeat and the crystal structure of the splicing module. Today, I'm just going to concentrate on the core and its interaction with the trap subunit. So as for the lobe in TF2D, the core module builds on an octamer of histone folds, which include a pair that is just intramolecular within SPT3H. And that kind of distorted histone-like octamer binds against the WD40 domain of the TAP5-like protein in Saga. Okay, so that's very similar, as I said, to lobe in TF2D. Then the N-terminal domain of TAP5 actually is engaged by another one of these Saga-specific subunit, which is the SAPT20H. That is what happens in the case of the human complex. In yeast, that domain of TAP5 is moved up 60 degrees, where it interacts with the heat repeat of the TAP6 subunit, actually blocking the binding site that in human is involved in engaging the splicing module. In TF2D, the N-terminal domain of TAP5 is actually moved 90 degrees in the opposite direction, giving rise to a different architecture for the otherwise conserved region. And as of course, this module is repeated, is present twice in lobe A and lobe B within TF2D. So the sub-T28, I told you, the N-terminal domain interacts within the core with the TAP5, but then it has a very long extension that contributes, that wraps around the trap subunit and through this latch that then ends on a C-terminal domain. So it contributes to the interaction with the core, with the trap subunit, which is otherwise quite robust. It involves most of the subunits within the core. But this is the case in human. Again, there's a difference with yeast in which that interaction involves fewer components of the core, and it has a cleft in the middle that makes that interaction more flexible. It lacks that long extension in the SPT20 that wraps around the TRA1 subunit, the homolog of TRAP, but it has an extension on TAF12 on the N-terminus that wraps following a different path. So interestingly, while the core, the scaffold that makes up the core is quite similar between human and yeast, to the point that it is actually quite obvious how these two can be aligned and superimposed, the differences end there, because this common core interacts very differently with the TRAP and TRA1 subunit. The human TRAP interacts with the bottom of the complex, while the yeast TRA interacts with one side of the complex, giving rise to an overall geometry that is very distinct between the functions of TRA1 engaging the activators and the core that then interacts with the flexible histone-modified units. As I said, TRAP1 interacts with transcription factors and is actually the site of human mutations that give rise to disease. They include the serine 721 to phenylalanine that is predominant in melanoma, but also a number of other mutations that give rise to neurodevelopmental diseases. We can now map those in the structure, see which ones are exposed, maybe affecting interaction with these activators, which ones are buried and maybe affecting conformation of this module. I just want to point out two among those which disrupt interactions between the TRAP and elements in the sub-T20H. And each one of these give rise to a neurological disease, and they involve regions that are not present in the yeast structure, explaining why they wouldn't give rise to neurological disease in yeast. In any case, this is what I wanted to tell you about SAGA. I want to tell you now about NUA4. In this case, we've studied the yeast complex, and of course it has the TRA1 subunit that I already talked to you about. It has a core that is distinct, very distinct of different components with respect to what is in SAGA, that engages two regions that are flexibly attached, the tintin module that interacts with nucleosomes and maybe other factors, and the histone acetyl transferase module itself, which includes the active subunit, the transferase subunit, the SA1. So looking at the structure by cryo-EM, there's a hub again of well-defined density, including the TRA1 and the core, while the tintin and heart are flexible. So we only are able to see a number of the subunits because due to flexibility. Those that we see pretty well. We have a resolution of 3 angstrom, and that I'm showing you 3 views, one in which you can distinguish very clearly the shape of the TRA1. This orthogonal one that shows better the structure of the core, with this view now showing the predominant ARP module, including ARP and actin. And these are the models that this is the model that we're able to generate from it. So again, this exists in the context of a structure that has flexible element that we only see at lower resolution and higher threshold, and that include the tintin hat and the YAF9 component of the core that are flexibly attached. We know more or less what they are because we can follow some of the linkers, the flexible tethers into components of the core for all of them. Now, this structure comes after a structure that was published by Gan Kai in 2018, in which the assignments for the TRAB1, the ARP4, and the actin subunit were the same, but everything else is almost completely different assignment of subunit. That model was built at lower resolution with a structure, a density map at a resolution of about 4.5 angstrom. To make sure that we got the correct one, we used the cross-linking data from Calvin Yip that was also published like a month later in 2018, and map the distance, the crosslinks in both structures. And what we see is that for our structure, both intra and intermolecular distance fall within the 30 angstrom that is expected from the DDS crosslinker, while in the case of the previous structure, most of the intermolecular crosslinks are not fulfilled. They're just they're incompatible with the crosslinking data. In any case, in our structure, I want to describe how this TRAB1 subunit that interacts with activators then connects to the rest of the module. Here now I color differently the hit repeats, the fat and the pseudokinase domain that form the TRAB1 subunit. As you can see that the hit repeats are not involved in much connection. Most of the interactions involve the fat and pseudokinase domain, just like it was the case within SAGA. The most dominant module in the core is the ARC module that is dominated by the ACT1 and ACT4 subunit. This is just a quick schematic of the interactions. The ARC4 and ACT1 is the only one of they're the only ones of the subunits that we see in the core that do not interact with the TRAB1, sorry, and which interact with the other three, with EAF1 being a real hub of interactions. You can map through now coloring from N to C terminus red to purple, the path of this subunit that basically interacts as it goes along with all of the components, and that in its N terminus, which we don't see is flexible, is supposed to interact with the tintin module. So this is this protein is really the hub that holds the whole complex together. It contributes the HSA alpha helix to the interaction with the ACT1 and ACT4 that is shared with INO80 and SWEA1, where that particular helix is actually part of the subunit that gives name to the complex. In any case, if we now compare the NUA4 with the yeast saga from budding yeast from Praticrema, you can see again that where we align the TRAB1 subunit, you can see that the interaction with NUA4 of the core element in that complex is very extensive, while in the case of SAGA, that interaction involves mostly just STAF12 and the SPT20, and is much weaker in the case of the yeast saga, explaining that there's a lot more flexibility between the two. So just to finish, one of the things that we have done in collaboration working with EpiCypher was to see what is the nucleosome preference for the modules, for the HAT and tintin modules that we express recombinantly using their alpha assay. And we can see that for the HAT, It prefers the H3h4 methylation, with trimethylation being, more methylation being better, but ultimately it even prefers even more those nucleosomes that are also hyperacetylated on H3. This is not due to the acetylation contributing directly to the binding, because without the methylation, it's just kind of background binding, but probably due to accessibility of the tails, as previously proposed by Catherine Musselman. So for tintin, the results are a little noisier, but they seem to prefer hyperacetylation on H4 and H2A. So the tintin module has a certain preference for the modifications that the new A4 hat introduces in nucleosome. So just to end, we would like to propose a model of how NuA4 is recruited to their target nucleosomes, which we believe is probably very similar to how SAGA and other acetyl transferases, large complexes, are probably recruited. They have a core that to which both the acetyl transferase module and modules that recognize nucleosomes are flexibly attached. So the idea is the transcription factors bind to specific sites through interaction with the TRA1 subunit that recruit the complex, and within that neighborhood, the flexibly attached elements that can recognize different histone tails will select nucleosomes, and then the histone acetyl transferase will be able to recognize those and carry out the acetylation, and will be able to do it in a neighborhood that is proximal to the site of recruitment due to that flexibility. So I just want to thank Abby and Robert. They were graduate students that did in my lab when they did the work on TF2D that I showed you very early at the beginning. Now they're postdoc at Johns Hopkins at Northwestern University. Robert is part also of the team working on human SAGA. He worked with Megan when she was rotated in my lab on the purification of SAGA, which was improved by her now as a graduate student in the lab of my colleagues Xavier and Teach, with helps also from those labs. The work on the structure was done by Dominic, with some contributions from Chan Ling in my lab that I didn't have time to talk about. And Abby obtained the structure of NuA4 together with Stefan, who was an undergraduate in our lab and now is a graduate student at MIT. And with this, thank you very much for your attention. Hopefully you will have some questions.
Thank you, Eva, for a fantastic talk. The questions are already rolling in. And so I'm going to start with the one question from the attendees. I'm going to read it out. It's from Anirban Dasgupta. Anirban says, fascinating cryo-EM structures as always. I was wondering if the absence of sub-T20H C-terminal extension in yeast impacts the deubiquitinase activity of SAGA in any way. If yes, is there any other player which comes in to compensate the process?
Yeah, so that tether that is not visible, is not apparent in the yeast structure, really is just part of that interface with the trap complex, and I would imagine it has no effect on the acetyl transferase activity of the complex or the deubiquitinase activity that is on flexible elements on the opposite end of the complex, but it could have effect on the way the TRAP1 and TRAP subunit interact with different type of activators. So more with the recruitment and a specific activity at different genes.
Thanks, Eva. We have a question from Phil Cole, one of the panelists. Phil is asking, to stabilize the hat domains in the SAGA and NuA4 complexes to enhance electron density, have you tried adding inhibitors or megabodies?
We have not done any things like that. I don't know that inhibitors will have an effect. One obvious thing to look at is to look at them in the context of chromatin and engage with nucleosomes, but still we think is actually an important part of how this complex function, that the modules are flexible and that the side of engagement with specific sequences on the DNA is still allowed for activity on nearby nucleosomes due to this flexibility. So we don't anticipate that it's going to be a way of rigidifying them with respect to that core and the trusted unit.
Thank you, Eva. Looking for raised hands. Thanks again, Eva. We're good, and thank you for being the opening speaker. It's great to start with this kind of talk. Thank you. So now I'll introduce our next speaker, Vijay Ramani, who's speaking from UCSF, where the time is 8:57 a.m.
Awesome. Okay, I can get started? Can everyone hear me? Great. Okay. Thanks, Geeta. Thanks, Geeta and Danitza and Nevin for also the invitation here. Really excited to talk about this. I thought I'd just start by just highlighting the people who actually did this work. Everything I'm going to be talking about has kind of come together in the last 3 or so years since I started my lab, and in large part thanks to a really wonderful collaboration with Geeta's lab. This was work that was done by a graduate student in my lab, first graduate student Noor, first postdoc in my lab Karolin, and then an incredibly talented postdoc in Geeta's lab Laura, with some help from Siva Kasinathan and Maithili Keitavarappa, who helped with some of the analyses I'm going to show you today. So as the majority of the audience probably is not familiar with what my lab does, we are predominantly methods developers, and we develop new molecular methods to try to better understand the processes of transcription and try to understand how chromatin structure potentiates transcription. I like to think that in so doing, we're trying to combat a pretty serious observational bias that we all face as scientists. This is the so-called streetlight effect. And this kind of alludes to the fact that we as scientists tend to make biological discoveries and tend to look for new biology in areas that are illuminated by the methods that we use to describe biology. And so I like to think that we are trying to develop sort of new quote unquote streetlights to illuminate areas of biology. And then ultimately we're trying to address questions in the fields of chromatin and transcription that maybe aren't being adequately answered by the existing methodologies that are currently available to folks. And so today, in that regard, I'm going to talk about think about this metaphorical key that we're looking for, how are individual chromatin fibers structured at genome scale. And then I'll talk to you about a method that we published at the end of last year that we've continued to develop, which is really a new method that we use to study at genome scale the individual structures of individual chromatin fibers using next generation single molecule sequencing. And so the biology that I'll talk about is a topic that I think is exquisitely familiar to most people in the audience. And this is the problem of how chromatin remodelers, ATP-dependent chromatin remodelers, these large macromolecular complexes, how do they actually restructure intact chromatin fibers? And so as many in the audience will know, ATP-dependent chromatin remodelers can catalyze many different kind of activities on chromatin and using nucleosomes as a substrate. They can evict nucleosomes, slide nucleosomes, load nucleosomes. And obviously this is absolutely essential for many of the nuclear processes that are important for life itself. But there's a major problem that I'd like to highlight in terms of our ability to study chromatin remodeling and in fact, the ability of chromatin remodelers to structure individual oligonucleosomal fibers. And that is the fact that if we were to say, take an individual genomic locus or an individual genomic haplotype within a diploid cell, we could actually find cases where the same exact sequence moves through any of these processes, either the act of being actively transcribed, being repressed, or in fact, processes like repair. And many of the methods that we use for studying how fibers are structured, right? And I want to separate this from a lot of the really important single molecule resolved work that has been done on mono or even dinucleosomes. Many of the approaches we use to study individual fibers tend to suffer from an averaging effect, where we perform a biochemical assay like an MNase assay, digest up into many templates, many chromatin fibers, and then either through gel electrophoresis, but now more commonly through high throughput sequencing on the Illumina platform, we arrive at averages that are not necessarily reflective of the distinct biochemical and biological states that these templates go through. And so this to me and to our lab seemed like a pretty big problem to try to tackle. So we went towards trying to develop a new method that takes advantage of new technologies to try to really understand chromatin structure at the level of individual fibers. And again, before and after remodeling, and that's what I'll spend most of my talk talking about. I'll very briefly go through this method that we've developed in the interest of time. This is published, but our method takes advantage of a cutting edge, really new photonic technology that enables a single molecule real-time sequencing of very long DNA molecules. And this is a technology that's been marketed by a company called Pacific Biosciences, and it involves effectively immobilizing individual duplexes of DNA in what's referred to as a zero-mode waveguide, a very, very tiny hole. And this very tiny hole has been engineered to effectively detect single molecules worth of fluorescence. And so sequencing is carried out as a nucleoprotein complex that is a duplex DNA that has been immobilized with a Phi29 polymerase, is allowed to basically carry out a polymerization reaction in the presence of fluorophore conjugated nucleotides, and a movie is taken. And this is done in millions of wells in parallel and effectively sequences on long templates are decoded by virtue of the tracking of incorporation of one of 4 different fluorophores. And this actually now works quite well. This technology is a little bit old, but really it's only been in the last couple of years that it's become cost effective, and the quality, the accuracy of the resulting sequencing reads, as we call them, has become quite high. There's an interesting a wrinkle about this technology that I want to highlight that is really critical to how we make these measurements. And it's that this sequencer is so sensitive that the polymerase kinetics can actually be read out over the course of taking these movies. And so this is an old figure from a paper that really beautifully showed that if the PacBio sequencer were used to sequence templates that have M6DA installed on the sequence, the actual kinetics of the polymerase, the average time between 2 different base calls, is actually lengthened because the polymerase is now traversing this DNA modification. And this is obviously in contrast to an unmodified template. And so we wanted to develop a technique that could resolve the structures of long individual chromatin fibers. And so we then took advantage of the fact that the sequencer could natively detect some of these modifications. And so developed an assay that effectively uses an adenine methyltransferase to mark where naked DNA might be in the context of chromatin. And this was heavily inspired by a series of assays by the Jones, Clada, and Schubler groups predominantly, where they used GPC or CPG methyltransferases to effectively make this readout. And so as proof of concept with Gita's lab, we effectively created chromatin arrays on tandem repetitive Widom 601 DNA sequences. We then took a non-specific adenine methyltransferase. We created chromatin through salt gradient dialysis. We then took a non-specific adenine methyltransferase, methylated, and then the hope was that we'd basically be able to see nucleosome footprints. These are regions that won't be methylated because the DNA is in fact protected by histone DNA interactions. And so the idea here is we take these sequences that we've methylated, purify the DNA, we then put it on the sequencer. Hopefully this polymerase is going to stall, and we should see natively, just when we sequence these DNAs, shadows of non-modified sequences that are reflective of where nucleosomes were. And the spoiler here, and again, this is published, this works. So I'll show many pieces of data that kind of look like this. Every line here is an individual molecule. The purple is our computational prediction of whether the DNA is methylated or unmethylated based off of the change in the kinetic signature of how the polymerase sequenced this molecule. And we can see here really beautifully, this reagent is a bit funny. It's a non-nucleosomal array with 9 tandem repetitive full copies of the Widom 601 sequence. And then we accidentally ended up finding out that there's about 450 nucleotides of what ends up being plasmid backbone that will kind of in a stochastic way assemble nucleosomes. But we know from this kind of positive control experiment that this works quite beautifully. Okay, so this was published in eLife at the end of last year. We called this assay Samosa, the single molecule adenine methylated oligonucleosome sequencing assay. And we were able to show that this worked in vitro, but also predominantly in vivo, where we applied this to map chromatin structures in the K562 cell line. So I want to talk about in the next 13 minutes or so, unpublished data that really builds on this assay that Nora and Colin really championed. So I want to focus on a method, and I'm going to show some proof of concept data that makes the case that we can actually characterize the single fiber chromatin structures that are created through chromatin remodeling in vitro. And we're going to use SNF2H as an example, in part because of the massive amount of beautiful work Geeta's lab has already done in characterizing this catalytic subunit of the imitation switch complex. And then I'm going to shift gears a little bit and talk very briefly about how we can try to use the in vitro data that we generate to inform in vivo data that we collect from living embryonic stem cells that either express this catalytic subunit or have had this catalytic subunit knocked out. And again, we're going to use Samosa, the single molecule assay, to try to answer these two questions. Okay, so I'll talk about this first. So we showed through our WIDM 601 experiments that we could actually potentially use our Samosa assay as an in vitro platform for studying chromatin biochemistry. And so we wanted to formalize this. And so we developed, Noor and Karolin and Laura developed an assay that we're calling Samosa Chat. And this is short for Samosa, but we're going to test chromatin activities on assembled templates. Right? And so the workflow is illustrated here. We're going to, again, this is inspired by really pioneering work from the late Jonathan Whittam, but I think work that has been really nicely formalized by the Pew and Korber groups, where we're going to take native sequences from a genome. In this case, we're going to take mouse genomic sequences. We're going to clone them. We're going to prep them and then use these as templates for assembling chromatin of varying properties through salt gradient dialysis. In our case, we're going to test the property of density. And by density, I mean the number of nucleosomes per individual molecule of DNA, per template of DNA. And then we're going to either do some biochemistry or we're just going to look at the native sequence. We're going to Samosa footprint, and we're going to use our PacBio sequencer to detect footprints. And this works beautifully as it did work for WIDM 601. This also works for, in our case, a couple of murine genomic templates. We can, thanks in large part to a lot of computational work that I won't talk about by Colin, we have a neural net that can effectively classify stretches of unmethylated sequence. We see that these footprints, that I'm going to call them, really nicely correlate to mononucleosomes and dinucleosomes. But there's also some DNA breathing, which we would expect. And then also importantly, we can actually in a straightforward manner, calculate at single molecule resolution, how many nucleosomes, an integral number of nucleosomes do we expect on a template. And so the experiment that Laura and Noor did was they actually assembled chromatin of varying densities where we expect 5 nucleosomes per template, 10 nucleosomes per template, 15, 20, et cetera. And these medians of these distributions that I'm showing here really actually nicely follow the target number of nucleosomes we'd like to assemble on each of these templates. And so we really think we're pretty excited. We haven't done any biochemistry here, but we're actually able to create chromatin fibers on any sequence of interest. We see some really interesting patterns that I just want to take a couple of minutes to go through. Well, I'm going to visualize where nucleosomes are going as a function of sequence as a set of heat maps, where the y-axis here is the size of a footprint, and the x-axis is the position of a footprint midpoint. And so mononucleosomes are going to show up somewhere here as kind of balls of heat. And this is precisely what we see. And what we see is really quite nicely reflects sequence-directed nucleosome positioning. And so this is a random sequence from the mouse genome at varying chromatin densities. And this is an average of all the footprints from a bunch of fibers of this density. But we can nicely see these are regions that predominantly favor the formation of nucleosome core particles, and these are regions that do not. And this, as one might expect, tracks really nicely with primary sequence content. This is generalizable. We also see evidence of higher order footprints. We can say this is a dinucleosome, trinucleosome. These are stretches of DNA that are recalcitrant to methylation, and this does generalize. So I'm going to talk a little bit about. I actually won't talk much about this today, but we're going to call this sequence S1. But this is a totally different sequence S2, also from the mouse genome. Different sequence directed patterns, but still strong evidence for sequence directed nucleosome positioning. All right, we can begin to adapt more sophisticated algorithms to try to see how individual molecules might be interrelated. So every dot here, we're using an algorithm called UMAP to just effectively visualize the interrelatedness of each fiber that we've footprinted. Every dot here is an integral fiber. But what I think is really nicely, and all I'll highlight here, is that even with this unbiased computational analysis, just in 2 dimensions, we can kind of see patterns arise that are reflective of true biochemistry. In this case, increasing density of fibers. So we can footprint fibers that we've created, and we can analyze this data at single molecule resolution. This we're pretty excited about, and we think it reflects a lot of the powers of our new approach. But now we can do some chromatin biochemistry, right? And I'll go through this pretty quickly, but I'm happy to talk to people offline. This should hopefully be a preprint very soon, and there's going to be a lot of biology that I won't go into. But we can take these maps and take these fibers, and now we can do remodeling. So what we're going to do is we're going to, under saturating conditions, we're going to add SNF2H and ATP, and we're going to see what happens, right? And we were pretty excited by what we saw. Basically, in bulk, the remodeler, in this case, a remodeler that predominantly slides nucleosomes, it's not, we can quantify this, it's not really evicting nucleosomes based off of our results. In bulk, it's effectively overriding the sequence-directed constraints that salt gradient dialysis assemblies are facing, right? And again, this generalizes, we can do this for a couple of different fibers. Kind of more illustrating proof of concept, we can analyze this data at single molecule resolution. We can try to take these fibers, again, do something like UMAP to embed them in 2 dimensions, use clustering, and then ask, okay, if we have different catalytic conditions that go into these experiments, do they impact cluster distributions? We end up with maps like this. Each color here is a different cluster. We can facet out these clusters based off of different conditions where we say these are unremodeled. This is saturating conditions, but we've added ADP, or so this is, no, we haven't added ATP. We've added ADP in a couple of different conditions. We've added ATP in red here. I'll just highlight here. These are the remodeled fibers. And we can now begin to look at how state distributions are changing as a function of the kind of biochemical input. And so we're pretty excited about this. There's some biology that will be in an upcoming preprint that I really don't have time to go through. But I just want to visualize and show you guys what this data actually looks like, where we have different fibers of different densities. And again, here, we're using the same color scheme where purple is methylated, blue is unmethylated, every row is an individual molecule. But now we're going to add, in saturating conditions, SNF2H and ATP. We actually see the biogenesis, the formation of the chromatin arrays, these kind of well-spaced arrays that are typical of SNF2H remodeling. And we've done some quantification. I can't go into this, but our in vitro data really suggest a highly heterogeneous landscape of remodeling outcomes by this remodeler. And then a parameter that I'm happy to talk to people offline about, our remodeling outcomes are heavily influenced by the underlying density of fibers. And we're really excited about this result. I'm going to take the next 5 minutes to talk about how our attempts to kind of build on this platform, but now take some of the results that we are able to characterize with the Samosa CHOT assay and bring them in vivo. Can we actually try to understand how this remodeler is working on individual fibers in the context of in this case, murine embryonic pluripotency? And so we're benefited by the fact that we actually have a really beautiful experimental system, courtesy of Dirk Schubler's group, where we have embryonic stem cells that are completely devoid of SMARCA5 is the gene encoding SNF2H in embryonic stem cells. And then we also have a really nice overexpression experiment where the Schubler lab has taken this genetic background and then through a constitutively expressed cDNA overexpressed the catalytic subunit of SNF2H. And now we're going to just ask a bunch of questions, but really, where is fiber structure changing the most? And what we'd really like to see is is density, is underlying chromatin density playing a role, or is at least associated with the different outputs of this chromatin remodeler. So what Nora has done is she's taken these cell lines, she's performed an in vivo Samosa protocol. Our Samosa protocol effectively isolates oligonucleosomes from living cells. It uses a combination of light micrococcal nuclease digestion. So we know that the end of every molecule that we sequence is probably the edge of a nucleosome. And we can see really beautiful kind of nucleosome repeat length and well-ordered arrays. But what we see when we knock out the catalytic subunit of SNF2H is a global lengthening of the nucleosome repeat length in vivo. And this is highly consistent with what people have seen in fly, but also what Dirk has seen and what the Owen Hughes labs have seen in mammalian cells. But this is an average, right? And so the question then becomes, and it's something we were very interested in asking, we happen to know already from a lot of genetic experimentation and many other studies that the SNF2H remodeler actually has a bipartite output in cells, right? So SNF2H canonically, or I should say, imitation switch complexes repress chromatin, presumably by creating well-ordered arrays, but they also, in a subset of sites, particularly for CTCF sites in mammalian cells, seem to be essential for creating accessibility, which seems like an odd dichotomy, right? And so we wanted to investigate this and see if chromatin density might differ these sites where SNF2H is closing chromatin and versus SNF2H is creating accessibility. And we were benefited by the fact that we actually have a set of sites in vivo that are genetically dependent on SNF2H to stay closed, and a set of sites that are genetically dependent on SNF2H to stay open. And we were able to calculate this from published ATAC-seq data. And so these are the last couple of slides. We were able to show something I think that's very interesting. So what I'm showing here are distributions of absolute nucleosome density on individual chromatin fibers that fall within sites that are genetically dependent on SNF2H to stay closed. So when we lose SNF2H, these sites open up. And what we see, it's a small effect, but a very significant and very reproducible one, is that if we look at fibers that fall within these sites, in regions that, in cells that are devoid of the remodeler, these fibers are lower density. That is to say, they have fewer nucleosomes per kilobase. But when we add the remodeler back, the density increases. This is consistent with perhaps a model of condensation, where we're adding the remodeler back in vivo, and the fibers are becoming more dense. The effect size, again, is not very large, but this amount of DNA that is being protected is like about 30 nucleotides per kilobase, which if you think about the average enhancer, that's about enough space for a transcription factor to ectopically bind. Right? But this is very different from what we see in sites that are genetically dependent upon SNF2H to stay open. In these cases, the remodeler isn't actually changing local nucleosome density at all. And we were very excited to see this. So this suggests a model where the remodeler is creating accessibility, but it's not altering local nucleosome density all that much. And so we ended up looking at this for CTCF sites, and we're able to find a set of molecular states that the remodeler seems to be acting on to effectively create again, without changing nucleosome density all that much, create a state where in this case, the transcription factor CTCF could productively bind and access chromatin. And so I've moved through this quite quickly, but we think we're arriving at via our single molecule assay at a model that can reconcile the differences between SNF2H and imitation switch complexes closing chromatin and SNF2H containing complexes opening chromatin, and this might involve some SNF2H associated dynamics. So with that, I'm out of time. Again, this was work that was really beautifully carried out by Noor, Laura, and Colin, with help from Siva and Mike, who's an undergraduate. So happy to take questions. Thanks so much for your time.
Thank you, Vijay. Danitza, you have a question for Vijay? Go ahead.
Yeah, beautiful talk, Vijay. I really enjoyed it. So how to think on the molecular level of the instance where SNF2H is creating accessibility without changing nucleosome density?
Yeah, and I think this is a question that required me to go a bit more into the biology, and so I'll just say this now. So Geeta and others have shown really beautifully that SNF2H has a domain, this hand-sand slide domain, that interrogates flanking DNA, right? And so if there's not enough flanking DNA, then catalysis is not possible, right? And so you could imagine a set of states, a Goldilocks zone, let's say, of nucleosome density, right? Where if SNF2H is acting as a dimer, as some of Geeta's structures would indicate, then if there's enough flanking DNA on either side, then the remodeler could just ping pong nucleosomes back and forth without necessarily altering nucleosome density in a local way. And in this case, these dynamics would increase the, say, fraction of the time that, say, a motif like the CTCF binding motif would be accessible, and enable productive CTCF binding, right? This, in the cases of repression, perhaps there might be mechanisms by which other transacting factors can effectively, after SNF2H has effectively created or equalized linker lengths, perhaps another factor like HP1 could potentially cross-link and move nucleosomes close enough that catalysis is no longer available, and you shut off those dynamics, right? That's very speculative, but that's one way in which that you can kind of discern this between the same remodeler creating accessibility and then or reducing accessibility in both.
Thank you, Vijay. We have some questions in the Q&A box. I'll start with the first one from Zadira. Sorry if I'm mispronouncing the name. Zadira says, thank you for super exciting talk. As I understood, Samosa relies on the activity of CpG methyltransferases to map the nucleosome density positioning of DNA sequence. I wonder how accurately does Samosa map the nucleosome positioning of native DNA sequences that have low or no CpG sequences?
Hi, Zadira. This is a great question, and I went through this very quickly. So we don't use a CpG or GPC methyltransferase. This is actually something that just differs our method considerably from methods from Dirk's lab or from Jones or Clada groups. We use an M6A, an M6DAA, so an adenine methyltransferase. And so we're pretty excited about this because adenine methylation is incredibly rare in mammalian genomes. And so we like to think that we're probably not perturbing the chromatin landscape or perturbing the structural landscape all that much. Also, there's absolutely no crosstalk with existing epigenetic modifications in these organisms. So yeah.
Next question is from Anirban. He says, great talk on a very interesting method. Love the acronyms. This might bust open the Pandora's box of chromatin remodeling landscape. Do you think this assay might also be useful in scoring the interaction of chromatin remodelers such as SwitchNIF with major hat or other epigenetic writer complexes in vivo in terms of alterations in chromatin accessibility and dynamics?
Totally, right. So we're very excited in vivo, right? We're using a constitutive null model right now, right? But we have active projects in the lab where we're tagging different transcription factors, different hat complexes, right? With say degrons, right? Where we can actually get very acute chemical genetic perturbation and depletion of some of these factors, and then pairing that with Samosa, right? The other side of this coin is that to the extent that we can reconstitute some of this biochemistry, we can use Samosa, kind of like the Samosa Chot assay, and then see how the two match, right? It's kind of a top-down genetic perturbation approach, but then a bottom-up reconstitution approach, and that we're hoping will illustrate a lot about how these complexes and how these activities actually work.
And then the last question is from Jenna. Jenna says, great talk. How does the binding of transcription factors and other large DNA binding complexes affect the methylation you see? Or is the methylation repression specific to nucleosomes?
Yeah, I'm just gonna go backwards in one. Can I go backwards? Oops. Let's see if I can go backwards on this. Yes, so we can see transcription factor footprints. So this in this plot, right, in cluster 6, these footprints are nucleosomes. These footprints, when you see them, right, these very short stretches of protected DNA are transcription factor footprints. So we can classify effectively based off of footprint size, whether it's a nucleosome footprint or a transcription factor footprint. We haven't yet looked to see if we reliably see subnucleosomal complexes like hexosomes, but we're interested in doing that.
Okay, scanning for raised hands. I think that was all the questions. Vijay, thank you again for a great talk.
Super, thanks everyone.
And our next speaker, Sebastian, are you there?
Yes.
Our next speaker is Sebastian Deando, who will be talking to us from Uppsala, where the time is currently, if I'm not mistaken, 6:24 p.m. It's evening time. Thank you for joining us, Sebastian. All yours.
Thank you, Gita. I'm going to share my screen. All right. Okay, great. So I would also like to thank the organizers, Gita and Daniele, for the invitation and for putting together this wonderful meeting and list of speakers. So it's a great honor and pleasure for me to give a talk today. And I thought that unlike the title that I originally gave, that it might be better in the context of this seminar or symposium to speak about very recent structural work for my group instead of the single molecule imaging approaches that I typically talk about. So apologies for the last second change here. So I'm probably going to have to skip this slide almost entirely, since this audience doesn't really need much of an introduction into chromatin. So I just want to remind ourselves that obviously transitions between different chromatin states are really quite important in allowing the cell to switch on and off certain subsets of genes. One way this can be done is through the use of these chromatin remodeling enzymes that we've already heard a lot about, so no introduction needed there. It's really important that the activity of these enzymes is controlled very carefully. And I want to talk about that. I think what is really quite fascinating is that now that we have a bit more of an understanding of sort of this core mechanism of nucleosome translocation, that many of these remodeling enzymes can actually simply shift the histone optimer along the DNA. For example, SNF2H that we've heard about and that Gita has done a lot of beautiful work on. This is a figure that I've stolen from a review that Greg and I recently published, where basically we sort of try to synthesize a little bit of the basic nucleosome sliding mechanism as maybe we understand it now. I think Greg is going to talk a lot more about that, so I'm going to leave it at that and won't go into any details. I just want to sort of highlight that it's quite exciting for me to then, knowing that sort of there is maybe this basic core mechanism, that we now need to understand how different accessory subunits and accessory domains that virtually all of these chromatin remodeling enzymes have can impinge on that basic mechanism so as to even alter remodeling outcome or at least regulate the activity. I want to give you one striking example of a remodeling activity that needs to be regulated carefully. But this is generally, of course, true for many remodeling enzymes. Misregulation can lead to severe disease states, including many different types of human cancers. And so today I want to talk about ALK1, amplified in liver cancer 1, which is a chromatin remodeling enzyme that was shown early on to play some kind of role in tumor formation. So this is very early work from a group in China, where they took ALK1 overexpressing cells and they injected them into hind extremities of nude mice. And that stimulated tumor formation greatly. And then in 2009, there were two seminal papers that established ALK1 as an important factor in the early DNA damage repair. More recently, work from Simon Bolton's group, and my group has contributed some to that work, established essentially that ALK1 is an attractive target in therapeutic intervention in cancer. And I should mention that there were also 3 other papers that showed very similar results, including Gulia Tyminski's group, Andreas Ladurna's group, and then Roger Greenberg's lab that published very similar findings. And I think the main conclusion from these papers is really that ALK1 inactivation sensitizes to clinical PARP inhibitors, and it also confers synthetic lethality in homologous recombination deficient cancer cell lines. And so that then, of course, makes it quite interesting for us to understand how ALK1 is regulated and how it's switched on and off. And I want to show you just very briefly the domain architecture. So like all chromatin remodeling enzymes, it possesses this ATPase motor here, in this case at the N-terminus, and it's split into an N-terminal and a C-terminal ATPase lobe that then together can hydrolyze ATP and essentially help generate motion or couple ATP hydrolysis to motion. And then at the C-terminus, we have a macrodomain that's connected by a linker region that's quite long. And this macrodomain is really what makes ALK1 unique among chromatin remodelers, because it can help to recruit ALK1 to sites of DNA damage. So one common hallmark of DNA damage is that early on, poly-ADP ribose chains are formed, so-called PAR chains, for example, by poly-ADP ribose polymerases. And then if we have these PAR chains, ALK1 is recruited, and it's known that this recruitment not only binds the remodeler where it's needed, but it also activates and releases the sliding activity of ALK1. And in the interest of time, I'm not going to go into the details, but I just want to sort of briefly summarize this because it sets the stage for what I'm going to tell you in just a bit. My PhD student, Laura, a while ago came up with sort of this finding that the macrodomain of ALK1 serves as an autoinhibitory module, and she derived, using low-resolution structural approaches, a structural model where we could see that the macrodomain is abutted against the C-terminal ATPase lobe. And that basically stabilizes this inactive conformation of the ATPase motor where the two lobes are splayed apart, and so not competent for ATP hydrolysis. And that's good in the absence of DNA damage, so there's no wasteful ATP hydrolysis. And then when DNA damage occurs, the recruitment of ALK1 by the macrodomain to these part chains, as we could show, releases the macrodomain and sort of displaces it away from the surface of the ATPase motor. And that seems to prime the remodeling ATPase to then eventually show this motor lobe closure, presumably when the remodeler is then recruited to the nucleosome. And the question is, of course, which aspects of the nucleosome, which features are recognized, and also, of course, how does the recognition of these features then switch on and release the remodeling activity of ALK1? And for this, of course, it would be nice to have a structure. In particular, we looked at cryo-electron microscopy studies of complexes between ALK1 and the nucleosome. And I want to really acknowledge two young scientists in my group, especially Guillaume Gaulier, but also Luka Bacic. And Guillaume Gaulier is an expert cryo-EM person in our group, and he has been trained by Karolin Luger or in her lab, and I'm very happy to have him. So he's really been amazing in this project. So what Luca and Guillaume did is they decided to use nucleosomes that are asymmetric. So they have a 10 base pair short linker on one side and then no linker DNA on the other side. And it's important that this 10 base pair linker has a 5' free phosphate group, because that's required to recruit this PARP2 enzyme, which can introduce pyrylation on the nucleosome. So it generates part chains from NAD, but it only does that in the presence of this histone pyrylation factor 1 or HbF1, as we know also from Ivan Matic's lab and also work done by Ivan Ahel's group. And Karolin Luger also has done some very nice work on this. And so using this in combination then allowed us to generate these nucleosomes with part chains. And then once ALK1, the full-length enzyme, and ADP-BEF is added, we can basically then you see that there's a complex formed, and this is really what ended up on the gel. Look at this using vitrified sample, and then here you see that this finally then gave rise to these individual particles, and we could collect a large data set. And one thing that was quite obvious from the very beginning is that there was a large amount of conformational heterogeneity in the data set. But there was also a smaller set of particles, just I think around 5,000 to 6,000 particles that adopted a stable conformation. And I'm going to show you first a reconstruction that is done exclusively with this smaller set of particles. You see that the resolution is at least sufficient to see good secondary structure. And that then allowed us to place a molecular model into this. And I'm showing you now the ATPase motor here in pink. So dark pink is the C-terminal ATPase lobe, and then in light pink, the N-terminal ATPase lobe. And what I want you to see here is that basically this ATPase motor of ALK1 bound to the nucleosome shares two features that we've seen already previously in nucleosome-bound structures where the ATPase motor was in an active conformation from different labs. And one of these features here is that the ATPase motor engages the superhelical location 2. This is, of course, a prototypical interaction site for remodeling enzymes on the nucleosome. And you see also that the ATPase motor lobes are in this sort of very closed and active, maximally active conformation. And then another feature, which I think is quite interesting, is that this histone H4 here, if you look at this, the N-terminal tail, it protrudes, of course, from the nucleosome disc, and it makes interactions with the C-terminal ATPase lobe. And that is something that has been seen by several other groups, that interaction between the histone H4 tail and the C-terminal ATPase lobe is required for releasing maximum ATPase activity of these remodelers. So this is maybe not too surprising, And I should mention that this active conformation of the ATPase motor is very, very similar to a structure that was recently published while our manuscript was under review from Su-Chen Chen's group, where they've visualized, or they've basically bound the full-length ALK1 enzyme to the nucleosome by cross-linking. So they don't have a paralated nucleosome, and they use cross-linking to generate a stable complex. And they saw essentially this maximally active conformation, and it's very similar to what we're seeing here. But because we didn't resort to cross-linking, we could also see a whole range of other conformations on the nucleosome. And to make a long story short, what Guillaume did to analyze this is he used the CryoDragon software package to sort of look at plausible intermediate states between these two extremes that I'm showing you here. So we see that there is this maximally on state that I've just shown you, where the two ATPase lobes are really tightly gripping the DNA at SHL2. And then there's a whole continuum of different structures. And I'm just showing you an intermediate resolution sort of experimental map here. And you see that in this extreme case here, there is really a very wide open ATPase motor conformation. So this looks exactly like the structural model from 2017 that I've shown you earlier, where basically the ATPase is completely off and auto-inhibited in this off state. And it's only very loosely attached to the nucleosome, but I think it's kind of neat that it's still attached to the nucleosome. And this also looks very similar to a crystal structure from Su-Chen Chen's group that was published in the same paper while this manuscript was under review. And this actually looks very much like the crystal structure of the isolated ALK1 enzyme in the absence of nucleosomes in terms of the overall configuration of the two ATPase motor lobes. And then, of course, there is a continuum of states in between. And in many of these more loosely associated states, we see that the C-terminal ATPase motor engages the histone H4 tail of the nucleosome. And then we also see interactions between where we believe the histone H3 tail is, that's paralated with the macrodomain of ALK1. And then we also see interactions between the ALK1 remodeling enzyme and the nucleosome acidic patch. And that, of course, is a very prominent interaction surface. So just to remind you, the acidic patch is formed by these negatively charged residues on the surface of the nucleosome disc. And it's used by many nucleosome interacting protein machinery as sort of this interaction surface. What we wondered is actually whether ALK1 might utilize the acidic patch as well. And this is from an earlier manuscript where we noticed that in cross-linking coupled to mass spectrometry experiments, a number of cross-links were formed between the linker region of ALK1 and the histone core of the nucleosome. And that led us to look more closely to this into this ALK1 linker region. And what we noticed is that there is a striking sequence similarity between the N-terminal portion of ALK1 of the linker, which we then call the regulatory linker segment, and this so-called LANA sequence. And this LANA peptide is derived from the Kaposi sarcoma virus. And the interesting thing is that there is a very nice, beautiful structure of the nucleosome bound to the LANA peptide by Karolin Luger's group. And of course, we were inspired by that and thought that maybe the linker might also interact with the acidic patch. And just to make a long story short, this is indeed the case. So I'm showing you an experimental map of the nucleosome, and we've cross-linked a regulatory linker segment, a very short segment, but nonetheless, it actually allowed us to look in more detail at this interaction between the ALK1 regulatory linker segment and how it might engage the acidic patch. And in particular, there is this key residue here, this arginine 611 residue that inserts itself into this depression on the surface of the acidic patch. And that has been seen also in the LANA structure from the Luger lab and in several other situations where proteins have been visualized bound to the acidic patch of the nucleosome. And so then, of course, the question is, what happens if we perturb these interactions? And we can do that by looking at nucleosomes that are either not paralated, so in the absence of the histone paralylation factor 1, or nucleosomes that can be paralated by, in this case, PARP1, when HBF1 is present. And we can use a readout for the nucleosome remodeling activity that is FRET-based. So this is actually an assay that was originally developed by Gita Nalika's group. And in this case, we have a Cy3 donor fluorophore on the histone octamer and a Cy5 acceptor fluorophore on the DNA, in this case at the end of the DNA. And if we initially place the nucleosomes close to the DNA end using a 6:1 positioning sequence, then we can use FRET as a readout for the relative distance here. And then as remodeling progresses by ALK1, the histone octamer will be moved to more central positions, and we go from high FRET to low FRET. And we can look at that and then derive initial nucleosome sliding rates from this. And this is what I'm showing you here. At increasing ALK1 concentrations, the initial remodeling rates. And there's not much of a surprise if we look at wild-type nucleosomes and ALK1 full length. Without nucleosome pyrrolation, we basically have a canonical Michaelis-Menten curve. But what was really surprising is that if we added now HbF1 into the mix and we allow for nucleosome pyrrolation, now we have this very strange deviation from the Michaelis-Menten canonical saturation curve, where we have this first initial increase and then a decrease actually of the initial sliding rates until we reach saturation. And I think the most plausible explanation for this actually is, let me move this window here a little bit. Oops, okay, is that if you look at the nucleosome substrate in these assays again, it's very asymmetric. So we have a short 10 base pair linker on one side and then a 63 base pair linker on the other side in these assays. And what you can imagine is that if PARP1 binds now to either side, of course it can bind and it can autopyrrolate itself potentially, but only when it binds to the short linker DNA end can it actually pyrrolate the histone H3 tail that emanates from the nucleosome pore here. But on the other side, PARP1 is too far away to actually pyrrolate the nucleosome. So essentially we obtain an asymmetric pyrrolation of the histone octamer, which would then, of course, also lead to an asymmetric recruitment of ALK1, predominantly to this short linker DNA side of the nucleosome. And that would lead to the nucleosome basically being remodeled towards the center of the DNA. And that's also what we observe in this initial nucleosome sliding rate. And then, of course, as we start titrating in more and more ALK1, first we saturate this binding site because it's a high affinity binding site, because the macrodomain, of course, can bind to these part chains, but it can't do that on the other side of the nucleosome, so on the other symmetry-related SHL2 location. And so then only once that high affinity binding site is saturated, we would start recruiting ALK1 also to the lower, weaker binding site and start remodeling from that SHL2 location, in this case then in the opposite directionality, so again towards the shorter linker DNA. And what that does is that at higher ALK1 concentrations, we start seeing this reduction in the initial nucleosome sliding rate, despite the fact that we increase ALK1 concentration. And I'm almost done. Yes, so what does that then mean for the interactions between ALK1 and these epitopes that I've shown you before? Well, if we start perturbing these interactions, for example, by mutating the linker here, this canonical arginine anchor, you see that there is a decrease essentially at all ALK1 concentrations that we look at in comparison to the situation where we have a wild-type full-length ALK1. And the same thing is true if we look at the other side of the interface. So if we mutate the acidic patch, so these are the APM nucleosomes, and then use wild-type ALK1, again, we have a reduction at all concentrations. And the same is also true if we mutate or truncate the histone H4 tail from the nucleosome. So if we essentially abrogate the interaction between the H4 tail and the C-terminal ATPase motor lobe. I don't have the time to show you these data, but we repeated these experiments, but instead then looking at ATP hydrolysis rates. And that closely mirrors what we see here. So this effect is really mostly due to these nucleosomal epitopes being important in stimulating the maximum ATPase rate for nucleosome sliding by ALK1. I want to just finish with a very speculative sort of model of what could happen in a local chromatin context. So if there's a DNA damage happening here, it could leave a short linker DNA end that is short enough, like in our in vitro assays, to then lead to a pyrrolation of this proximal histone H3 tail. And that would lead to an asymmetric recruitment of ALK1, preferentially to this side, which of the nucleosome, which is now the high affinity binding side. And that would lead to then a preferred remodeling direction, where overall this initially short linker DNA is then extended by being pushed out of the nucleosome, so that that would sort of very locally relax chromatin and potentially allow DNA repair factors to come in and fix the damage more efficiently. And with that, I would like to highlight again Guillaume, Gaulier, and Luca, and also Laura, Klaus, Anton, Mao, Sophia, that have also contributed to this project, and especially my long-term collaborator Simon Bolton in his lab on this project, and Mario Halic for very interesting discussions on data analysis using this CryoDragon package, and Mark Scale for the cross-linking coupled to mass spectrometry data. And I thank you for your attention.
Thank you, Sebastian. That was an awesome talk. I see Greg has raised his hand. Greg, do you want to unmute and ask your question?
Sure. Yeah, that was really great. That was fantastic talk, Sebastian. I guess my question was about your view about the acetylation on the H3 tail being stimulating. I guess I was trying to understand how that connects to the linker binding to the acidic patch, because it seems like it's quite far away in the model that you have. And I just didn't understand, maybe I didn't quite understand.
Sorry, Greg, do you mind repeating the question? So are you thinking about the macrodomain binding to the site that's paralated, to the histone H3 tail that's paralated preferentially? Is that what?
Yeah, I guess what I'm getting at is that in your cryo structure, it seems clear that it's the tail that the little lightning bolt is pointing to here. I guess we'd call that the, right by the ATPase, the closest region. And I guess my question was, and maybe I just missed this, but that's supposed to release the linker. I guess I didn't quite understand if it's really preferential for it to be. Is it interacting with the H3 tail in your model while the macrodomain is with the ATPase, or can they be uncoupled? So my question is, if the macrodomain were bound to the other tail, the one that's labeled H3 tail, would that still allow the ATPase motor to be activated? I guess that's sort of ultimately my question. Does it matter which side it's on?
Right. I honestly don't know. It might. So I think it's a very interesting question in general in terms of the molecular details of this. I think what we would really need to do, and we've been discussing this quite a lot, is to actually do some single molecule experiments where we can ideally have even an asymmetric,
Right.
By definition, asymmetrically paralated nucleosome of defined composition, and then really look exactly at what's going on, where the binding happens, and in what remodeling direction. So the short answer is we don't know, I think. Yeah.
Right. Okay, great. Thanks.
Thanks, Sebastian. There are two questions in the Q&A. One is from Anirban. He says, wonderful work. Do we know if any other PTMs help recruit ALC1 to nucleosomes individually or in concert with PAR chains?
That's a good question. I'm not sure. I mean, with ALK1, we've always thought sort of the macrodomain as being sort of the main recruitment module, and then of course the ATPase motor binding the nucleosome. We don't know. I'm sure there is. I mean, the more sort of I think the field learns, the more complex things are, and I think that's definitely a possibility. I'm not aware of any particular modification.
A question by Wilson in the Q&A. Great talk. In the Zuchen structure, the linker interacts with acidic patch via ARG614. What could explain the differences in the APM epitope?
Yeah, that's also something that we've noticed, and I think that from everything we've seen in the sort of the data set as a whole from cryo-EM is that there's a lot of flexibility going on. And so there are many different possibilities. One thing could be that, there is not one canonical arginine anchor, but actually if you look at the linker, there is a number that are quite close in close proximity to each other. And so it's entirely likely that, it doesn't always have to be the same one. I do know that cross-linking tends to fix and bias potentially for the one versus the other configuration. But on the other hand, our structures was derived from a short peptide, and that's obviously very different from having the entire ALK1 structure. So it could also arise from that. But my feeling is that there is a lot of flexibility, so it could potentially utilize different arginine anchors at different times, and all of these could work and serve the purpose essentially.
Vijay has a question. Vijay, do you want to unmute and ask?
Okay, yeah, sure. I also typed it, but yeah. So Miguel Bito's lab had shown several years ago that poly-DP ribose could actually serve as a nuclear ATP source through this enzyme NUTX5, right? So I'm wondering, like, could ALK1 or remodelers in the context of DNA damage generally be actually consuming PAR chains to fuel remodeling? Have you guys thought about that at all?
No, in fact, I've never heard about it. That sounds very cool, to be honest.
I'll send you the PMID. Yeah.
It's Miguel Beato's lab, you said.
Yeah, exactly.
Yes. No, that's very exciting. I mean, it is true that these nucleotides looks at the end of the day they're quite similar. So sounds very interesting. Mm-hmm.
Cool.
There another question in the Q&A box from Wasan. Great talk. Is the length of DNA from nucleosome core to PARP2 binding site sorry, I'm trying to, I think there's a grammatical issue here. Let me start again. Is the length of DNA from nucleosome core to PARP2 binding site essentially short, or would it work with a longer overhang too in vivo? That's the first question. I have another question. You want to go take that first?
Mm-hmm. So I don't think I can give a quantitative answer to that, but I think that the proximity to the, if I understand it correctly, to the nucleosome core does matter, and so we haven't really studied this in detail. So one could do this very systematically, but I could imagine that at some point you end up being essentially in a position too far away from the nucleosome where it would be sterically less favorable to have efficient PARylation on that side.
Great. And the second part of the question is, does the observed continuum in the ATPase conformation arise due to lack of cross-linking, or could there be something stabilizing it?
So from everything that we've seen on ALK1, I think it is quite dynamic. I think that this makes perfect sense because, there are all these different epitopes that it needs to sense, and we don't know exactly in what order, but it's probably constantly, sensing those, and this would probably argue for, in addition to our cryo-EM data set, that there's a lot of flexibility. So my guess is that cross-linking in this case for Su-Chen Chen's work helped to sort of nail down and stabilize one particular conformation. And that's the one that is maybe in the ensemble the most populated one, which is the active state.
Great. So we are now, we reached the conclusion of the first half of this morning. So we're going to go into break, and we will resume at 10:15 a.m. Pacific time with Karolin Luger. See you all back in a few minutes. Have a good break.
Thank you.
In the mountains, further high. Boulder?
Boulder. Yeah.
Karolin is speaking to us from Boulder, where currently it is 11:15 a.m. All yours, Karolin.
Thank you. Well, thanks, Gita, and organizers for this fabulous meeting. I'm actually not on a mountaintop, unlike what my background suggests.
We can't hear you. You'll have to, I think, something with the volume.
I'm unmuted.
Yes, we can hear.
Huh, that is weird. We had that before.
Still can't hear you.
I am unmuted.
I can hear fine.
Oh, I was on mute. Sorry, Karolin.
Okay.
So
Good.
embarrassing. Very
So we're all good?
Please continue.
We're all good. All right. Well, thanks, Gita, and thanks for organizing this fabulous meeting. I will continue with the theme of this particular session and explore this fundamental question posed by Roger Kornberg many, many years ago. What happens when an irresistible force meets an immovable object? And of course, something's got to give. And so what happens when a polymerase impinges on a nucleosome? Now, if you think about it in mechanistic terms or mechanical terms, if you will, some histones have to be evicted. And that poses several challenges. First, we have to prevent the non-productive interactions between histones and DNA and RNA, because histones are positively charged and DNA, RNA is just there and negatively charged. So that'll make a mess of things. We'll have to also make sure that there's an ordered and prompt deposition of first H3h4, then H2H2B in the wake of the polymerase to prevent cryptic initiation of polymerase. And we have to stabilize partially disassembled particles and kind of prevent things from just flying off and diffusing away. And then finally, this all has to be coordinated with the polymerase. And there's a large group of proteins called histone chaperones that are responsible for these tasks. They're structurally not related. They're involved in histone safekeeping, coordinated nucleosome assembly, and there's no ATP hydrolysis. And we haven't really talked about this group of proteins so far, but of course, what's on everybody's mind after the beautiful talks this morning by Sebastian is chromatin remodeling factors. And these are responsible for histone displacement, nucleosome disruption, exchange, and these are all ATP dependent activities. So today I'll tell you two brief stories about from one of these boxes, from the blue box and from the pink box. And so let's start out with the quintessential histone chaperone called FACT. I've kind of renamed it, much to Danny Reinberg's chagrin, I think, into facilitates all chromatin transactions, because it really reflects the pleiotropic roles that this complex plays. Consists of two subunits, multiple interactions with histones. It assembles and disassembles nucleosomes during transcription, repair, and replication. It's very conserved. And it also stabilizes partially assembled nucleosomes. What I find particularly intriguing is that cancer cells appear to be addicted to FACT. And so undifferentiated cells, as well as cancer cells, have rather obscene amounts of FACT. In cancer cells, it can be up to 1-to-1 ratio of nucleosomes to FACT. And we can, in the end, speculate a little bit why that might be the case. And so we were just simply asking the fundamental question, how does FACT work? We're not very sophisticated in this respect. So our lack of understanding or confusion on FACT function really doesn't come from a dearth of structures. There are a large number of structures of individual domains of FACT, but in this sense, we're a little bit like the blind man feeling the elephant, and depending on which part of the structure you look at, you might arrive at different conclusions as to what this complex is doing. And so it would be really important to have a structure of FACT doing whatever it is it's doing. And so fortunately, the cryo-EM resolution revolution came to our rescue. And so in a very fun and productive collaboration between Bridget Carragher's lab and Sheena Darcy's lab, we managed to tackle this question. And this was spearheaded by two fabulous postdocs in my lab, Yang Lu and Kira Zhao, who are both on the faculty market right now. And so if you see their applications, you should just grab them both and you won't regret it. All right. So in order to solve this problem, we needed to form a reasonable complex. All good cryo-EM starts with good biochemistry. This was really a difficult problem to work with, but through some tricks, and I can explain to you if anybody's interested why we had to do this weird assembly system where we first had to pre-bind FACT with histones H2H2B and then add them to a tetrasome consisting of 79 base pairs of DNA and H3H4. Finally, we managed to get a good complex. You can see here there's two bands here, and this actually resulted in two distinguishable complexes in cryo-EM, and this is ready for prime time. After a lot of trial and error, things aren't always as easy as Eva makes them sound. So after a lot of trial and error, we got some grids that were workable, shall we say, and then with the help of Bridget Carragher's WIS team in particular, some of her fabulous grad students and postdocs there, we managed to obtain 2D class averages, and that allowed us to solve the structure to a workable, shall we say, resolution. This complex fact sitting on top of a partial nucleosome resembles kind of a unicycle with the 2 dimerization domains of the 2 subunits forming the saddle, and then the 2 middle domains forming the fork going down on either side of this tetrasome wheel, if you will. And then we have H2A, H2B dimers. In this case, it's only one, forming the petals. I should say that despite our best effort, and this seems to be kind of a recurring theme, there's parts of the structure that we cannot see because it's too disordered. And this includes an N-terminal domain of SPD16 and a large region including a DNA binding domain of HMG. And we can also discuss why we might not be able to see that. So just to summarize the salient features of this structure, what did we learn from this structure? First, and probably most surprisingly, is that FACT has an extensive DNA binding surface. And if you look at the underside of this saddle, you can see it's very positively charged. And of course, it fits perfectly well on our very negatively charged tetrasome wheel on the DNA. The tips of the fork of our unicycle are basic, and that fits well, are acidic, and that fits well with binding to the basic proteins. This was a little bit surprising because we could show in previous work that FACT neither binds nor disassembles properly formed nucleosomes. And so it seems that when a nucleosome is healthy and well folded, FACT ignores it, but it needs to kind of be primed by a partial, by seeing a partial nucleosome structure, and it also needs to be primed to release some of the autoinhibition that we can do by histone H2H2B dimer binding. This DNA binding interface was confirmed by HDXMS done in Gina Darcy's lab by Naifu Zhang. It was very gratifying because they had no prior knowledge of this structure. And when they sent us the residues, the amino acid residues that were particularly protected upon complex formation between FACT and the tetrasome or the partial nucleosome, they mapped almost exclusively to this interface here. So this was a very, very nice confirmation of the structural data. The second interesting finding that had been anticipated by others already is that this C-terminal domain of SPT16, which is very acidic and kind of very noodly and disordered, kind of pretends to be DNA, and it protects the side of this histone H2A, H2B dimer that would normally be occupied by DNA. And so in a side view of these structures, actually in the beginning at very low resolution, we were confused because it really did look like a complete nucleosome, like a complete wrap of DNA, which really wasn't possible because we didn't give it enough DNA. So this arm here kind of hugs the H2H2B dimer to the remainder of the structure. This was also confirmed by HDXMS, and we found increased protection. This also confirmed that the histone dimer was in its proper nucleosomal position, just tethered to the remainder of the structure through this DNA-like protein arm, this DNA mimic, if you will. SPD16, the middle domain, has 2 modes of interactions with H3h4, and this is also important to the function. This was a previous structure solved in 2016, a crystal structure of just the middle domain of SPD16 on a tetramer. And this structure is absolutely not compatible with what we see in our more complete structure. And things become really interesting when you start to morph between those 2 bound states. So if we start with our structure, just showing the middle domain, we're then starting to unpeel the DNA as a polymerase would, kind of coming along. And it will just simply knock the SPD16 middle domain and with it FACT off its perch on the DNA, and then it could assume its position on the tetramer. All right, and on the other side, this movement will actually help to dislodge the H2H2B dimer and further free up the DNA to let the polymerase pass. At least that's kind of our working model right now. Presumably, the H2H to be dimer would still be stuck to the C-terminal acidic domain. And so to kind of keep everything together and not let things float off. As is often the case in cryo-EM, we got two structures for the price of one. They're distinguished by the presence of a second H2H to be dimer on the other side of the FACT complex. And so this is a complete nucleosome, if you will, just devoid of the complete DNA, whereas this is a hexosome. And what you see upon binding of the second dimer, you see quite a profound change, conformational change of the middle domain, a translocation rotating upwards to make space for this. And this is again seen much better when you morph between the two states of FACT. And you can see here these conformational changes that happen upon adding the second dimer are pretty much restricted to one side of the complex, whereas the blue side on the other side of the tetrasome kind of stays the same. So this whole conformational dynamics we think is really important for FACT function. And so we have a tentative model that we're trying to test. It's not that easy. In which RNA polymerase unpeels DNA from the H2H to be dimer surface. FACT only binds to a nucleosome that's kind of already a little bit disoriented with the help of ATP-dependent chromatin remodelers, presumably. FACT then straddles the nucleosomal diet and protects the H2H to be dimer through its CTD, and that prevents the DNA from rebinding, so it can be worked on by the polymerase. And we think it also tethers the H2H to be dimer during subsequent steps. We then get a switch to the second binding mode through the mechanisms that we don't really understand fully. That results in a destabilization of the dimer on the other side. And then presumably the advancing polymerase dislodges the FACT middle domains from the DNA. And then FACT will kind of hop over on to bind to the tetramer instead. We don't quite understand how FACT tethers all the histones. We assume it kind of holds all the histones together and also maintains some kind of a foothold on the DNA while the polymerase works on this. And then in a reverse pathway, FACT promotes the reassembly of nucleosomes. FACT is not a very good de novo reassembling factor, but it can reassemble from this partially disassembled state. This was really very nicely confirmed by recent work from Lucas Farning when he was still in Patrick Kramer's lab, where he showed the structural basis of nucleosome transcription mediated by CHED1 and FACT. And quite interestingly, what Lucas could find is that in their structure of a polymerase with a nucleosome with FACT, the perch of FACT was yet in a different location compared to what we see. They used yeast FACT and we used human FACT. We don't really think that is the difference. We think really this whole FACT gymnastics and its ability to engage with nucleosomes in different ways is important to its function. And that's also why these structures are so difficult to determine, unless you're people in Eva's lab. I think these guys are they just rock. All right, so let's talk about the second group of proteins that help a polymerase navigate the nucleosome. This was the sole work of John Markert with some help from Cody Zhao. John was really interested in this very little studied remodeling factor called SMARCAD1. Now, as you guys have heard from Sebastian, chromatin remodelers use ATP to promote either nucleosome spacing or sliding in the I-switch family, CHED1 family. They can promote histone eviction, the switch SNF family most notably. And they can promote histone variant exchange or just histone exchange, for example, the INO80 family. SMARCAD1 falls into this latter category, at least functionally. And I should mention that its yeast and POMB names are FFT3 and FUN30, in case you know them under these names. So what does SMARCAD1 do? Like many remodelers, it's very hard to pinpoint. It's involved in many biological processes, transcription, replication, DNA repair. It's a very simple remodeler in terms of domain structure. It has one single protein chain, one single subunit, has two Q domains that maybe bind ubiquitin, and then it has two ATPase domains, just like any self-respecting remodeler should have. They're also called helicase domains. So what's the structure? What's the mechanism? How does it interact with the substrates? To answer the first question, John used a very nice FP assay that we're using a lot in our lab to show that FACT binds to nucleosomes and to DNA with very similar affinity, about 160 nanomolar. The binding of FACT of SMARCat to a nucleosome is regulated by phosphorylation. SMARCat binds to nucleosomes only when it is dephosphorylated. And John could show that there's a number of phosphorylation sites at the N terminus of the protein that have to, the phosphate groups have to be removed to allow binding. There's no binding, the fully phosphorylated form. And there's also papers showing that phosphorylation regulates recruitment to DNA damage. And this is something that we're really interested in at the moment. SMARCat, like any ATP-dependent remodeler, hydrolyzes ATP. ATP hydrolysis is much better stimulated by nucleosomes. This is also really not that surprising. It's nice to see this verified. And so then the next question was, what does ATP hydrolysis accomplish? Does SMARCat1 exchange histones? So John kind of developed an assay first shown by Gita's lab a little further, using trinucleosomes reconstituted with fluorescently labeled histones, and then adding acceptor DNA that is shorter and enzyme, and then start the reaction with ATP, and then test whether we get short fluorescently labeled mononucleosomes. And we can run these out on a gel and monitor fluorescence, and you can do this in a time course here. This is kind of a representative gel. The trinucleosomes are all in the well. Mononucleosomes run down here, and the nice, so you can see that SMARCat actually does exchange histones. It takes them from the trinucleosomes, puts them onto the 147 base pair DNA. But we can also do rate constants. And what was really interesting is that reproducibly histone H3h4 and H2H2B are exchanged at the same rate. And obviously, this process requires ATP hydrolysis. So because this happens at the same rate, John was wondering whether SMARCat can bind all histones simultaneously and kind of transfers the histone octamer as a whole, which is really not something that I would have predicted to happen. To show this, he used a FRET assay where he fluorescently labeled H3H4, H2H2B with donor and acceptor respectively, then added SMARCat, so no DNA present, and then look for high FRET, which would occur if SMARCat kind of brought the two histone pairs together. And indeed, that's what he finds very reproducibly. And the apparent KD for those histones to come together, I don't know what that is, for the two histones to come together is at 1 nanomolar. Oh, I think that was my time signal. All right. Can SMARCat assemble nucleosomes? Yes, it can. And this is a simple nucleosome assembly assay from scratch, de novo. And you can see here that SMARCat has quite robust nucleosome assembly activity, no ATP required for this. John then, of course, was interested in the structure, and he managed to get a low-resolution cryo-EM structure of this complex SMARCat bound to a nucleosome with asymmetric DNA. We can only see the two ATPase domain, the Q domains, and the intrinsically disordered domains are not visible, but that was enough to actually build a model from a homology model from other remodelers, because the ATPase domains are quite conserved. Now, we could capture two nucleosome states. This one to the right here might be an artifact of cryo-EM grid preparation, but John then also took a page from Guillaume's, his former lab buddy, Guillaume's book, and tried cryo-dragon. And even in our main structure, you can actually see there's quite a lot of structural heterogeneity. And so this is kind of the thing with cryo-EM structures. It's just a gift that keeps on giving. He managed to put the model into these morphs, and what we suspect is happening, that there's a 30-degree rotation down the face of the nucleosome, and that results in the interaction of SMARCAT with the H2B alpha C helix that we think we can also see by cross-linking MS. So this is relatively new data. I just thought I'd share that with you. And cryo-dragon is, I think you can have a lot of fun with it. It might be a little bit dangerous just because you might be walking down a rabbit hole, but it's a super useful and fun program. The interaction of SMARCAT with nucleosomes is unlike that of any other remodeler. Here is iSwitch, which is its closest relative, and SMARCAT sits kind of much closer to the diet and engages one arm of the DNA on this side here. And this was shown, summarized in a very nice review that John published in TIPS a little while ago. Interactions with the H3h4 tails, despite our low resolution, we see clear interactions with the histone tails. And I just finished with saying there's different roles for the different tails, and I don't have time to show the data. The H4 tail is required for ATP hydrolysis, the H3 tail is not. But histone exchange requires both the presence of both tails. So a tentative model for SMARCAT function, SMARCAT engages the nucleosome. ATP hydrolysis leads to the partial unraveling of the DNA and eventually the entire removal of the DNA. SMARCAT holds on to the entire histone octamer, we think, and then reassembles them on a fresh piece of DNA. All right, with that, I'll finish, and I'll acknowledge John, who is the man behind SMARCAT, and then Young and Cody, who are my fabulous fact team with Bridget and Sheena's help. And I'm happy to answer questions. Thank you.
Thank you, Karolin. Thank you for an awesome talk. We have one question from Anirban. Anirban says, wonderful story and great presentation. Work from the Wahlberger lab connects FACT to UBP10, which is a deubiquitinase module for H2B K120UB. Could FACT recruitment to nucleosome be initiated by H2B ubiquitination or other associated cross-talking Ptms? Could this possibly explain how FACT prefers slightly destabilized nucleosomes?
Yeah, absolutely. And this is like a whole can of worms that we are thinking of opening, looking at how FACT gets recruited via post-translational modifications. So that's a really good suggestion. Thank you.
Many more facts to be discovered about. Greg has a question. Greg.
Yeah, maybe you already said this, Karolin, but have you looked to see if the SMARCA-D1 sort of synergizes with some chaperone, since it seems to be able to do this on its own, but I wonder if they would help stimulate rates or final amounts?
Yeah, we haven't done that yet, but that's also something, a direction in which we're moving, because nobody works in isolation. So teamwork, sure, I think, is also a thing among chaperones and remodelers.
Great.
Eva has a question. Eva.
Hi, Karolin, great talk. I'm just fascinated by the fact that the two examples that you showed, whether using ATP hydrolysis or not, can work in both directions. They both can take things off the DNA, put them back on the DNA. And, I don't really know what it means. And I suppose in the case of ATP, there is a directionality going in one direction and back that is determined by that, the nucleotide state somehow. For FACT, when there's no ATP hydrolysis, what is it that does it? What allows the thing to switch from one? Is that regulated by something else, or it's just happening in both directions? And it is, I don't know, the polymerase that determines in which one is happening.
No, I agree. It's quite puzzling. I think for FACT, it's actually it's intuitively quite obvious, because it doesn't hydrolyze ATP. And we actually think FACT just kind of cruises around looking for for sick nucleosomes and then makes them whole. And possibly that's why it's so prevalent in cancer. And it's actually also known that when you delete FACT in yeast, you get a phenotype that's more consistent with an nucleosome assembly defect, unlike what's been thought previously that it's mainly there to plow the way for the polymerase. With SMARCAT, we don't really know. In vitro and in vivo are two different things. There's quite a bit of evidence to show that it's consistent with an in vivo function, that it can also go both ways, but it's less clear what would switch it. But we do know that the ATP hydrolysis is only required to remove the histones, not to put them back on. Because that might be thermodynamically favored, like it has these histones, what should it do with it? It just dumps them back on DNA.
Okay, we'll end with one last question from Nicholas Prescott. Nicholas says, beautiful work. You might have alluded to this briefly, but have you tried looking at SMARCA1 binding to ubiquitinated nucleosomes to see if that allows you to resolve any density on the CUE domains?
Yes, excellent question. And we are hiring. And actually what I forgot to say, there's postdoc positions available. Good segue, thank you. And you can hang out with Princess Krios, there she is. And we actually get fabulous resolution of nucleosomes. It's just too bad that we already know the structure of the nucleosome. And so these remodelers are kind of a bear. It's not because our instrument is inferior, it's just those are kind of very heterogeneous complexes. So yeah, that's a really super interesting question that we want to pursue, given the manpower or woman power.
Thank you, Karolin, again for a fantastic talk.
Thank you, guys.
I will now move on to the next speaker. It's a pleasure to introduce Greg Bowman, who's speaking to us from Baltimore, where the time is currently 1:43 p.m. Greg, all yours.
Okay. Well, thanks very much to the organizers and Gita in particular for the invitation to join in this symposium. For the last few years, we've been trying to understand how chromatin remodelers reposition nucleosomes on DNA. And what I'd like to tell you today is about some recent work on a published structure where we now can see how the remodeler is deforming the DNA and in fact changing the twist, which we think is really a core component of how this DNA shifting occurs. So we've already heard about, again, the job that remodelers do, and I'll just want to focus in on the question of how they actually are doing the sliding of nucleosomes. So they bind to this site we've already heard about before, SHL2 or superhelix location 2, sort of 3 o'clock on the nucleosome. And they're basically DNA translocases, so they can pull the DNA on towards them, and this has the effect of repositioning nucleosomes locally, as we've already heard about as well. Now, this is a unidirectional reaction, and yet they have to be able to move the nucleosomes back and forth. But this is possible because the nucleosome has this twofold symmetry. So there's an SHL2 site also located on the other side, and action of a motor in exactly the same way will just, of course, reverse that reaction or move it back the opposite direction. Now, these, again, as Sebastian pointed out, these have a very classic architecture of two sort of halves of the ATPase motor. These are part of a helicase superfamily called superfamily 2 of ATPases. And some of these translocate, non-remodeler helicases translocate along DNA and RNA substrates. And there's a really nice mechanism for that translocation. It's been termed the Inchworm Mechanism, first proposed by Dale Wigley, where the ATPase motor oscillates between an open state that's nucleotide free and one that's closed when it's bound to ATP. And we were interested in understanding how does this mechanism of translocating along a single-stranded substrate, how can that be translated to remodelers and how they shift a DNA duplex around the histone core. So our first indication for this came a couple years ago when a student of mine, Jessica Winger, was doing some tests looking at DNA changes when CH2-1 binds and found that, in fact, there was a change about 30 base pairs away. So here we're looking at the interactions of histone DNA with a cross-linking reaction. And this is the product of that cross-linking. And we could see that the cross-link was shifting by about 2 nucleotides when CH2-1 was added. And this was only happening in the nucleotide-free state, or ADP, but not with ATP mimics. This also, same type of behavior was observed at intervening cross-linking sites, but not on the other side of the binding site. And so from this, we concluded that what seems to be happening is that when CHG1 is binding here, it's distorting the DNA and allowing the nucleosome to absorb about a base pair, and that would correspond to the shift in all of these cross-links. Now that shift has to be a corkscrew-like motion, and if you corkscrew one side of the DNA and not the other, that will result in untwisting of the DNA. And so we proposed that, in fact, it's the untwisting of DNA that's responsible for sort of absorbing this extra DNA locally on the nucleosome. And I won't get into the data of it, but we also looked at the other states, the ATP mimic states, and found that, in fact, those reinforce the canonical twist of nucleosomal DNA. So we propose that, in fact, it's really just sort of a twisting, untwisting mechanism at this SHL2 site that seems to be responsible for pulling DNA on and then presumably shifting it past. And that resonates with a very old model proposed by Ken Van Hold back in the 80s, where he proposed basically exactly what I'm showing you here, this idea that the nucleosome can locally absorb a base pair or so of DNA and then give it up, and as a mechanism for thermally ratcheting around the DNA around the histone core. That idea, unfortunately, was sort of cast aside early on in the early 2000s by their modeling field, because there were some results that suggested that the DNA doesn't rotate when remodelers shift the DNA. Unfortunately, that turned out to be wrong. And that was shown really beautifully by Sebastian Deando in 2013. He showed a single molecule that, in fact, it's moving one base pair at a time. And so to keep all of the histone DNA contacts, it would have to have a corkscrew rotation with the translation. So what's really exciting to us is how this idea has sort of begun to take on new directions that weren't expected. So structures that were solved just a couple years ago helped to support and extend this model. And these are from Zhucheng Chen's group at Tsinghua University. He has a few cryo-EM structures in the nucleotide-free and ADP-bound states, where he sees the ATPase motor is engaging with the DNA and actually causing, he can see extra phosphates here in the structure. These are about 3.4 angstrom resolution. And but what really was surprising about these structures is that it's only absorbing extra DNA, or sort of pulling the DNA on one of the two strands. So he saw an extra phosphate on the red strand here, but not on the yellow. And so this is really, again, unexpected and very exciting, to find out what the remodelers are doing. We've been interested for a long time in the structural changes and distortions that the ATPase might impose on the DNA. And we've teamed up with Jean-Paul Armache at Penn State University. He's an expert in cryo-EM, and he's solved this amazing complex between the CHG1 remodeler and the nucleosome, and gotten it down now to 2.3 angstrom, sort of unbelievable resolution. And at this resolution, we can see just unprecedented detail of the geometry of the DNA. As you can see it not only every base and identify what that is, but also its geometry, how it's oriented. See the backbone really quite beautifully, and as well, of course, many side chains and how they're interacting and sort of holding the DNA. And this structure is a nucleotide-free structure that's bound, and again shows this expected distortion, where it's allowing it to absorb a base pair or absorb a nucleotide on one of the two strands. And to show you how it's changing the DNA, I want to just show you this morphing transition from a closed state to an open state. So the closed state with CHG1 was solved by Patrick Kramer's group and Tom Owen Hughes. CHG1, they're virtually identical. They're bound at SHL2, as we've seen. This is sort of looking through the histone core where it would be, but without the histones being there. So this is the SHL2 from the histone viewpoint. And I'm going to transition this now to the open state where it absorbs the DNA here, and just to show you how that is affecting the DNA geometry. So the first thing that we can tell from this is that as the ATPase motor opens up, it's actually shifting by one phosphate along the backbone here, along the tracking strand. And that's exactly what is predicted and expected from the inchworming model proposed for single-stranded translocases, that they sort of take a step forward. And what, of course, no one expected until Zhucheng's structures is that, in fact, in the process of doing that, it ends up pulling the tracking strand by one phosphate. You can see also here that the other strand, the guide strand, is actually not moving with respect to the motor. And of course, because we have some binding site changing in position and others that are not, the DNA has to distort. And this distortion, what's really remarkable is that it turns out to be transformed towards an A-form-like geometry in this open state. So let me explain to you what I mean by that. So here I'm showing you different parameters, and these are key parameters that distinguish A-form and B-form geometry. And I'm showing you them with respect to the nucleosomal DNA, starting at the dyad here on the left and moving all the way through base pair 45. The yellow bars are the structure that we have now that's bound to CH2 under the ATPase. The gray bars, which look sort of green here when they're overlapping with yellow, those are the other side of the nucleosome, the unbound side, and also all of these traces are other nucleosome alone structures, one that was in the same data set and other structures that have been published. So we can see that, and these are all 601 nucleosomes, that these all have a very similar parameters, but the binding of the ATPase motors, of course, shifting some of those from where they would normally be. Let me just show you what these are. So first of all, if we think about B-form DNA versus A-form, there are a couple of classic differences. So B-form has its base pairs that run through basically the center of the helical axis, whereas A-form is much more of a hollow appearance because the base pairs have shifted out. They're sort of more on the edge. And so that shifting here is what we call X displacement. And so we can see that's very negative, as you would need to be in order to reach an A-form geometry. As well, there's also inclination, right? So what's the tilting of the base pairs relative to the helical axis? A-form is classically tilted, whereas B-form is virtually perpendicular to the helical axis. And then the third parameter that's really quite important is called slide here. Slide corresponds with the shifting of one base pair stacked on its neighbor. And so you can imagine, so this base, for example, stacked on its neighbor here, it's shifted off compared to that of B-form DNA. And we can see that that also is perturbed, but interestingly, it's only at the site of the binding, and it's really just very localized. So it has this very local distortion of DNA, and this is really the spot where it's becoming the most A-form like geometry. Now this, I guess this also brings us to a different way of thinking about the DNA movement. It's not so much that it's grabbing one strand and pulling it, as the sort of single-stranded translocase models might have suggested, but instead it's binding to the duplex and just distorting it. And by making it this A-form-like geometry, A-form naturally has sort of one phosphate shifted on one strand relative to the other compared to B-form. And so it's just this geometric change that's responsible for causing the DNA to move. And again, we can understand that as a local sort of untwisting of the DNA in that site. Now, if we look at this from the remodeler point of view, and here what I've done is taken these two structures, I've morphed between and just superimposed them on ATPase lobe 2, the blue half here. And you can see from that the DNA, the backbone is in a pretty similar placement, but of course the underlying DNA is quite different. It still has all of the base pairs are still made, but you can see that the orange strand, because it's been shifted by one, this A-form-like geometry, it's causing that base pairing to be perturbed. But again, the remodeler is really recognizing just the phosphate backbone. So it can recognize these two different structures. And in fact, we think that it's probably the fact that the remodeler has very specific phosphate binding sites that forces the DNA to be sort of ratcheted a full one nucleotide ahead. Now, when we compare this to so I'm showing you now is comparison of the DNA as it's changed when it's bound to the ATPase versus when it's just the nucleosome alone. And this was, again, a nucleosome in the same data set to 2.6 angstrom resolution, shown in gray here. And I'm superimposing these now based on the histone core. So we could see that the DNA that's bound to the remodeler is sort of bulging away from the surface. And this is where it's accommodating that extra nucleotide. And this, although it's away from the surface here, interestingly, once it leaves the binding site, it rapidly gets back on track again. But as it's absorbed an extra nucleotide, it's sort of offset or sort of staggered in a way. All the base pairs are still kept, but now we can see this inclination that we saw before is now much stronger than it was in the nucleosome. What's interesting is that one turn of DNA later, we see now a distortion of the other strand, the guide strand, and this is absorbing here an extra nucleotide compared to the nucleosome alone. This then causes an inclination basis the other way, and that gets things sort of back on track again. So the inclination it's virtually the same outside of this, after the second distortion. And this brings up two important ideas. One is that by making this A-form-like geometry, it's of course, as we've seen, like pulling just one of the two strands, but the consequence is that rather than pulling a full base pair on, the other half of the base pair has to be introduced somewhere else. And we see that here at SHL3, and between those two sites, it will require that the DNA is going to have this type of distortion, or what we believe is a stress or a tension. And the second point is that this type of distortion that we're seeing at SHL3 resembles what's been seen before at SHL2 and SHL5 in many crystal structures, where SHL2 and SHL5 are known for picking up or giving off just a single base pair. Here we're seeing that happen just on one of the strands, and we think that this is happening because it's under stress or strain, and likely during remodeling, this is happening at other positions around the nucleosome. Sebastian Deando has some really beautiful single molecule 3-color fret where he looked at the entry side DNA being pulled on at the same time as the exit side DNA being pushed off. And in those experiments, he saw this ATP dependent delay between the entry being pulled on and exit being pushed off, where we think that the nucleosome itself is absorbing extra nucleotides. And we think that this SHL3 type of structure is likely happening around the nucleosome just when the remodeler is sort of inducing strain throughout the nucleosomal DNA. Okay, so the ATPase motor seems to prefer to bind to sort of a canonical twist of nucleosomal DNA when it's in this closed ATP bound state. But then once it opens up, it forces the DNA into this distortion, which resembles this A-form geometry, pulling just one of the two strands. And as I've just said, that sort of sets off this inclination that distorts the DNA in the downstream region. Now, to pull the other strand, we think simplistically, it just needs to close back up again. So binding to ATP is ultimately going to move the guide strand within the binding site. We think there are clearly some interesting steps and structural states here that we haven't yet seen that are important for transitioning from one to the other. But what this is showing us is that, in fact, unexpectedly, the remodeler is breaking up, sliding a base pair into two separate steps, where it first slides one strand, and then by changing back again, it can pull the other strand. We don't really fully understand the necessity for why it's doing that. It could be that it's sort of ratcheting it up in terms of energetic levels, but also one possibility is that it's giving it a directionality in sliding. Okay, so one other thing I want to say in the last few minutes that I have remaining is the unexpected discovery we had in looking at the density. We first saw this sort of at 3 angstrom resolution, that there was this tube of density going along the backside of the nucleosome. And again, I'm just saying the backside because when the motor is acting here, this DNA is being pushed off. So we call this the exit DNA, and we identified this as the N-terminal region of CH2-1. So we call this checks for CH2-1 exit side binding. And I just want to point out that we, with collaborating with Stephen Freed at Johns Hopkins, he's an expert in cryo and cross-linking mass spec, and was able to identify that in fact this was the CHG N-terminus. And subsequently the structure, the resolution improved such that it was quite obvious that that's what it was. But early on, the cross-linking mass spec was really essential for that. And here's what that looks like. So it's making this interesting interaction. Those familiar with the nucleosome will see this is the alpha N helix of H3 with basic residues, and it has acidic residues that are complementing that. And then, as we've seen and Sebastian's pointed out, it's interacting with the acidic patch. So this is, again, a very common place that many proteins, remodelers, and other factors will recognize the nucleosome. And it's using this canonical arginine binding to this acidic triad, another arginine here as well interacting with H2A, which is seen in some other factors, and then also a tyrosine that packs against this hydrophobic surface on H2A. And all three of these residues are pretty highly conserved in CH2-1 orthologs from yeast to vertebrates, and interestingly also found not just in CHD1, but in other CHD family members, 6 through 9. These have a very distinct biology, but the fact that they maintain this structure suggests that they're going to have a similar type of interaction with the nucleosome. Now, what's interesting to us is the fact that this is on the exit side. Gita's group has shown really beautifully that I-SWI remodelers and INO80 require acidic patch as an epitope in order to be active. One thing that we've found, along with Tom Mears' group, is that in fact, it's just the entry side acidic patch that I-SWI remodelers care about. And so again, with the twofold symmetry of the nucleosome, the exit side for one remodeler is the entry side for another remodeler. And so since this is so essential for ISY remodelers, we wondered whether or not this element could potentially interfere or slow down the sliding by ISY remodelers. Now, since these are both able to slide nucleosomes, we decided to dispense with the ATPase motor and just fuse this checks module onto the histone H3. And so when we look at sliding, we can see the classic signature, which is that the ISY remodelers will take an end position nucleosome and slide it to a more central location. This is a native gel, and so it migrates more slowly when the nucleosomes are in the center. When we give it instead, though, a nucleosome which has this core H3 fused to the checks module that replaces its N-terminal tail, we see that now sliding is really debilitated. So for ACF, it's about 3, I thought that was my data. No, it's about 50-fold slower here for ACF, and for SNF2H, it's at least that. We can't actually measure the rate because it's so terrible. And so that's telling us that, in fact, CH2-1 should be able to interfere and compete with the ISY remodeler. I don't have time to get into the data of this, but when we delete the checks region of CH2-1, then we see the sliding goes down about 2 or 3-fold. So it is affected, but it's not so dramatic as what's seen with ACF. So we think this might give CH2-1 the upper hand in competing with other remodelers that are acting in opposition to it. Now, one last thing before we go, and this actually fits really beautifully with everything that Karolin just talked about, which is to talk about what's happening during transcription. And so this brings up the possibility that this tail is not just involved in competing with other remodelers, but maybe it has to do with histone transfer. We have no data on this. This is just all speculative, but she pointed out a structure that Lucas Farnung had solved with FACT-bounded polymerase. He also solved a structure with polymerase and also CH2-1 bound. CH2-1, in fact, actually couldn't be bound at the same time for some reason, interestingly. And they didn't see checks in the structure, but this is where the checks region would be, and it's bound to this H2H2B dimer. And so to us, this raises this possibility that as polymerase is moving through and unwrapping the histones and H2H2B is going to be exposed, CH2-1 maybe help either stabilize or potentially transfer this to another histone chaperone-like protein to aid in the remodeling. And again, we're excited just to look in to see what role this may have outside of just nucleosome recognition and potential competition with other remodelers. Okay, so with that, I'll just end by thanking the people that did the work. So Jessica Winger really spearheaded and carried out the majority of the biochemistry, looking at the twist defects or looking at the shifting that CH2-1 does on the nucleosome, and had some help with Rob Lewandowski on that point. Ilana Nodelman did all of the biochemistry that I showed, this new unpublished work, prepared the samples for cryo-EM and did some really nice structural analysis. Jean-Paul Armache, in addition to, of course, analyzing the structure, really came through with this beautiful structure along with his student Cyan, and I think this is really going to be a landmark structure in helping us understand even more about how these amazing enzymes work. And as I mentioned just briefly, Stephen Fried also played an essential role along with his student Annalise in showing us that Czech was responsible for binding to the acidic patch. So with that, I'll end and be happy to take any questions.
Thank you, Greg, for a really thought-provoking talk. And do people have questions? While people are putting down questions, I just have a comment, Greg, and this relates to your description of how the field evolved in its conception of whether you need to twist the DNA or not. And in the old days, 10, 15 years ago, people would make nicks in the DNA and use that to say, look, there is no twist because it can dissipate. But your data beautifully shows that you can still explain how you can have nicks and still have this one base pair offset, right? So you don't, nicks are completely compatible with your data. Is that right?
Right, and that's an excellent point. And I think the problem is that, of course, with free DNA, nicks are able to, rotate and you eliminate all supercoiling. But the difference is that the nucleosome is really constraining the DNA. So even with these nicks, it's still, and again, many groups have shown this really nicely, that nicks are even gaps. You can take away one strand. I mean, there are some experiments where it's even missing, 4 to 10 nucleotides on one strand, and it's still able to slide. So it's kind of amazing what it can support. So,
That's great. Karolin, you can ask your question.
Yeah, that was beautiful story, Greg. Fabulous resolution. I'm just wondering, because it's known that these twist diffusion states can actually occur naturally, and they can be stabilized by factors binding and things like that.
Right.
What is the next step? So now it kind of binds to this distorted step, and then what? Like, what is happening next to cause remodeling, to cause actually sliding?
Right. So that's a great point. So the movie I was showing was transitioning between the open and the closed state, and it was just going back and forth because we just have this. And I think the problem is that I think that the beryllium fluoride ADP state, that there's actually many structures for, not just for CH2-1, different modelers bound. I think that presents sort of an endpoint after sliding, or the point before it's initiated. But there's a critical difference here, is that when it's opening up, the lobes are opening and it's sort of straddling and taking up more space on the DNA, right? Like a larger footprint. And then when it closes up again, I think what's happening, but we don't have data for this, is that it's closing up and sort of in a way walking forward. So it's actually inchworming forward on the nucleosome and still maintains a distortion. And then that just, I think it's this is speculation, but I think it's the distortion that we don't yet have beyond the apo state. So once it closes, that is encouraging the DNA to corkscrew to relieve that extra base pair, right? So I think we sort of have a half a base pair right now with the apo state. And I think that when it binds ATP, it pulls in a full base pair, but it's still trapped at SHL2. And I think that it's the relaxation of that that's going to be sort of forcing the DNA past the dyad. So that's the mystery that we don't yet have yet, is how that happens.
Because, as you know, you can actually stabilize these twist diffuse state with sequence-specific DNA binding reagents.
Right.
Okay, well,
yeah,
fabulous story. So
yeah,
we don't need to get into the technical details.
Sure.
It's beautiful stuff.
So one question from Sebastian in the chat. Sebastian says, "Hi Greg, very cool talk and amazing structure. Do you think it might be possible to visualize more than one or two single base pair twist defects on the nucleosome as potential remodeling intermediates?" Then he says, "I guess it might be hard to capture them using cryo-EM."
Yeah, that's a great question. I think, again, what the structures before would have suggested is that the nucleosome can take on, Tim Richmond has sort of suggested even you can have a 143 nucleosome. So you can, at each SHL2 and SHL5, you can take on or absorb one base pair. And I think what we think is likely happening is that it's able to absorb more than those two sort of past SHL2, more than just the SHL2 and SHL5 on the other side. But I think that under enough strain, it probably could populate other sites as well. So my guess is that it's going to be absorbing a single base pair sort of twist defect at one site, but who knows? It could be that it could absorb even multiple at one site, but I think it's more likely it'll be distributed and having sort of single insertions at multiple sites. But yeah, but we don't know. And I think cryo-EM is great for that. Again, as we've heard with all the dynamics, it's really just going to be getting tons and tons of particles to be able to see the different classes to see if it, but I think that eventually that will be shown. Yeah.
Great. We have two questions in the Q&A, and then we'll move on to the next speaker.
Okay.
We have a question from Hayden Saunders. Hayden asks, in your CHD nucleosome structure, are there any corresponding changes in the histone octamer structure, or is distortion only propagated through the DNA?
That's a good question. We don't really see any clear distortions. And I guess I'm particularly thinking of Mario Halich has some really beautiful intermediates that he's trapped with cross-linking that show sort of what you would expect of how it changes. And we don't see that. The nucleosome is interacting pretty canonically with the DNA at the histones with the DNA at all the other sites. So we don't see anything happening there. So my view is that it is really primarily happening through the DNA. That seems to be enough. But yeah.
Then we have another question from Anirban. Anirban says, awesome structures. I may have missed it, but does CH2-1 interact with Pol2 and the associated elongation factors, given that it interacts with ubiquitin-related nucleosomes?
Yeah, that's a great question. So it's known, Karen Arndt has done some really nice work to show that it interacts with RTF1, part of the path complex. It also interacts, it does bind to FAC, they interact together. and other elongation factors. There's not really been any structures of those. I think it's sort of like back to Eva's talk that there's a lot of flexible regions that interact, or they're intervening flexible regions. So it does bind to these other factors, but not as so much of a domain-domain interaction. But presumably those are also helping keeping it where it needs to be by the transcription, by the polymerase as it goes around the nucleosome.
Great. Thank you, Greg, again for an awesome talk. And I just want to clarify, I think the next speaker is Daniele Canzio, but Tatiana, are you switching to Daniele?
Sorry about that.
No worries.
Should I go next, Gita?
Yes, please share your slides. So it's a pleasure to introduce Daniele Canzio as the next speaker. And Daniele is speaking to us from San Francisco, where the time is 11:11 a.m. All yours.
Sorry, Gita, I'm just having a quick technical problem. Can you all see my screen?
We can see it.
Can you hear me?
Yep.
Great. So it's really a pleasure to be here. Thank you, Gita and Danica, for this wonderful meeting and for inviting me to speak today. So I really like to start by posing a fundamental question in the field of neuroscience. And that is, given that our brain houses billions of neurons that need to make trillions of connections, how do neurons really find their partners to assemble and wire a functional brain? So this is a really complex question of complex solutions that goes beyond my simple imaginations, and we certainly don't have an answer to this. But what we know is that essential to this process is the ability of neurons to be able to spread and innervate the brain, or in other words, arborize. And this was really apparent already from the work of Santiago Ramon Cayal over 100 years ago. And depicted here in these beautiful pictures really summarizing this wonderful arborization process of neurons, of different types of neurons. And what is central to the process of arborization is really a fundamental process. And that is the ability of neurons to be able to self-recognize and self-repel. So in other words, when branches of a single neuron come in close proximity, they have barcodes on their surface that allow them to self-recognize and repel. And this process basically allows, is the key, is the core of given the ability of these branches to basically innervate and spread. This process is conserved from flies to mammals and requires extracellular proteins that in flies are the DSCAM proteins and in mammals are the protocadherin proteins. Now the function of protocadherin proteins has really been shown beautifully by some elegant genetic studies from the independent genetic studies from the lab of the Sainz lab and the Maniatis lab a few years ago by looking at neurons in the retina of the mouse brain or olfactory epithelium, where these beautiful genetic studies deleted protocadherin genes in both systems and indeed showed what we refer to as self-avoidance phenotype, meaning the fact that branches from a single cell are not able to recognize and repel self anymore, but actually start interacting and collapsing. And this process, of course, is detrimental to not just the physiology of a cell, but the function of a cell. And disruption of protocadherin genes goes beyond simply the retina and the mouse olfactory epithelium, but we also know today that it's associated with several severe mental disorders, such as schizophrenia, autism, addiction, and cancer. So in part, the function of protocadherin genes really lies on the genomic architecture of these genes. So in humans, we have about 60 protocadherin genes. and each gene organized in 3 clusters that span about 1 million base pair of DNA. And each of these genes is represented here by a box. And each of these genes has its own nearly identical promoter sequence, here represented by this arrow. And each gene basically encoded a specific cell surface barcode. Now, as I mentioned before, arborization of neurons is really cell type specific. And so the brain evolved mechanism to generate different type of instructions that neurons utilize to spread and come together. And these instructions are encoded by, indeed, the protocadherin genes. So, for instance, olfactory sensory neurons, individual olfactory sensory neurons, chooses a different repertoire of protocadherin genes, such as, for instance, here, this red neuron, randomly or stochastically, chooses to activate alpha 1, alpha 11, beta 7, beta 16, gamma A1, and gamma 11. And this choice is very different from the yellow neuron, which is very different from the green neuron, and is different from the blue neuron. And so the fact that the individual olfactory sensory neurons can choose randomly their protocadherin promoters allows branches from the same cell to self-recognize and repel, but branches from different cells to actually come together in a particular circuit. So this is actually quite different from a different set of neurons. Serotonergic neurons, the one that are responsible for releasing serotonin in our brain, actually choose only one protocadherin out of 60 genes, and they always choose the same one in every single cell. And that basically allows branches from the same cell to, again, self-recognize and repel, but also branches from the same cell type to self-recognize and repel. And that mechanism allows these neurons to have a different set of instructions, and that is, instead of converging together, they actually tile and innervate the brain in a tiling mechanism. So it turns out that among the 60 genes, they always express and choose protocadherin alpha C2. So I'll show you with 2 different examples that here we have one locus, but many potential transcriptional profiles. So the really fundamental question is, what is the regulatory logic here? And the current view is that, in the field, is that perhaps cell type specific transcription factors really regulate cell type specific promoter choice. But what I would like to propose today is an alternative, slightly alternative model, where instead of different transcription factors, I'd like to suggest that different 3D genome structures actually drive distinct protocadherin expression. So this journey really started as a postdoctoral fellow in the Maniadis, in the lab of Tom Maniadis, where I basically primarily looked at the protocadherin alpha gene cluster in the context of olfactory sensory neurons. And recently proposed a model whereby stochastic choice of particular protocadherin promoters really requires that the promoters start as highly silenced. So the ground state of all the protocadherin promoters is characterized by, or the protocadherin alpha promoters, is characterized by DNA methylation and H3K9 trimethylation. And then essentially choice of a particular promoter really requires 2 steps: a random escape from this silent chromatin, and interaction between the promoter that escapes and the transcription enhancer that is specific to that cluster by 2 proteins known as CDCF and coenzyme. So this model really raised several other questions. And one of that was, what is the function of CDCF and coenzyme? And so when I started my lab as a group, we really started to investigate this model a little deeper. And we looked first at whether these same principles were indeed applied to the other 2 clusters, the beta and the gamma clusters. And so we turned back to our IC studies and looked at interactions between promoters, which are here located on this axis, and enhancers, which are also located in here. And so this is, for example, and the alpha specific enhancer, and these are the beta and gamma specific enhancers. And what we compared was olfactory sensory neurons, or FACS-sorted olfactory sensory neurons from the mouse brain, from wild-type neurons, and neurons that actually had a conditional knockout, RAT21, which basically ablates the cohesin complex. And so what we noticed was that the contacts between the enhancers and the promoters here in this axis were not only gone for the alpha cluster, but also for the beta and the gamma cluster. And here, this is the wild type, and here they're gone in the mutant. So while this wasn't that surprising, what was really surprising to us was when we looked at RNA-seq. So when we looked at our RNA-seq studies, we actually noticed that not only by virtue of losing enhancer promoter interactions, we were losing expression of all protocatenin genes. One gene in particular was actually upregulated, and that turned out to be protocatenin alpha C2. And this transcriptional profile that we saw in olfactory sensory neurons missing the cohesin complex was very much the same transcriptional profile that we observed in serotonergic neurons. So this was bulk RNA-seq, but then we asked, is this also occurring at a single cell level? Meaning, do single olfactory sensory neurons now choose primarily the alpha C2 promoter versus all the other promoters? And this is our single cell RNA-seq data from olfactory sensory neuron that indeed confirms that choice of a promoter is completely ablated, meaning that while in the wild type case, you can see in every single row being a different cell, a cell is able to choose a promoter in a random manner. In olfactory sensory neurons missing the cohesin complex, choice primarily goes to this promoter. So our data were suggesting the following hypothesis, that cohesin really governs competition between protocatenin promoters. And removing of cohesin ablates the ability of olfactory sensory neurons to choose stochastically their promoters of choice. And instead always choose by default the alpha C2 promoter, like serotonergic neurons. So to understand really what was the function of the cohesin complex, and so the genome organization function and choice, we then turn to ask the following question: what is the role of cohesin in mediating promoter choice? And to do that, I first want to introduce what we know today about cohesin and its function in genome organization. So the field has been proposing the idea that cohesin, this ATPase complex, uses the energy ATP to translocate along DNA and pulls DNA through its ring. And in translocation in a direction, in either direction, it stops, but whenever it finds this other protein that acts as a boundary element called CDCF. And so CDCF usually binds to specific DNA sequences here in red and blue that are characterized by a low level of DNA methylation. And wherever CDCF is bound, cohesin would then stall and promote contacts between different regulatory elements, for instance, an enhancer and a promoter. So at this point, we wanted to ask, is it that cohesin actually regulates choice of protocatenin promoters via DNA loop extrusion, also in this context? And so to do this, we set up to do a very simple experiment where we actually introduced a boundary element ectopically within the cluster. And so we generated a mouse where we inserted 3 CDCF sites within the locus that were meant to block cohesin translocation and cohesin flux. And when we did that, we actually learned by doing IC that indeed the block was working. Cohesin and CDCF were binding to our ectopic CDCF sites, and that was forming an ectopic DNA loop. But at this point, then we asked what happened to promoter choice. now that we actually insulated and created an external loop. And to our surprise, what we noticed was that actually choice became distance biased, meaning the promoters that were farther away were still chosen with the same sort of probability, but the ones that were closer to our ectopic boundary element were actually chosen with less frequency. And indeed, this data really suggested that there was a strong correlation between RNA expression and contacts between enhancers and promoters. So at this point, we went back to the drawing board and proposed that indeed random escape from random choice of protein promoters requires 3D genome reconfiguration by DNA loop extrusion and cohesion activity, but we were really not sure about what was the function of this mechanism. And we remember that indeed part of the sort of mechanism choice requires escape from a silent chromatin state or at least one of those promoters. And so we asked, is it possible that DNA loop extrusion actually facilitate DNA demethylation of the chosen promoter? So to do that, we set up another test, which is if you need an answer promoter interaction to actually demethylate the promoters, well, what if we actually delete or mutate the answer, the prediction would be that choice is actually altered because DNA loop extrusion won't be able to actually promote demethylation of a particular promoter. And so here we engineered two new mouse models, one where we deleted the HS5 enhancer, and one where we inverted the HS5 enhancer, meaning inverted the CDCF sites. And it's known that inversion of the CDCF sites, basically in this context, doesn't store cohesion translocation. So it basically acts as a deletion. And when we did that, after generating these mice, we then turned back to this question and looked at DNA methylation of specific promoters. And consistent with our hypothesis, indeed, we found that methylation of CDCF binding sites was actually altered in mice where the HS5 enhancer was deleted. And here I'm showing you just three different independent CDCF binding sites for three different promoters. And you could, I hope you can appreciate how the CpG element that is highly conserved in all of these sequences is actually hypermethylated when the enhancer is gone versus in wild type systems in context. And if that's true, that methylation indeed is altered, then choice should be also altered, so mRNA expression. And in fact, when we compare wild type olfactory sensor node to deletions of the enhancer or inversion of the enhancer, we notice again that they're consistent with our RAT21 data. The choice of which promoters to express was completely altered, where now all the stochastic promoters were off, and the only promoter that was chosen was the default alpha C2 promoter. So I like to conclude by propose a working model where DNA loop extrusion in this context actually defines the probability of protocatein promoter choice by mediating their demethylation, their stochastic demethylation. We certainly don't know how, but we would like to propose that this is a really interesting model to generate cell surface diversity. And perhaps the implication of this model are at least twofold. First implication is about brain wiring mechanisms. So as I mentioned before, perhaps a mechanism to actually generate different type of instructions that are specific to cell type innervation patterns is not by regulating transcription factors, but perhaps regulating and tuning the activity of cohesin on chromatin, where in olfactory sensor neurons is required for demethylating promoters such that they can be randomly chosen, and in serotonergic neurons instead is not. And so in this case, the choice always goes to the default alpha C2 promoter. But there is another sort of implication to this model, which is actually even more broad, that goes beyond the brain wiring mechanisms, and actually looks into potential models by which stochasticity is used to generate different type of cellular identities. And protocadherin promoter choice is just an example of that. But recent work from the old lab and others have also suggested DNA loop extrusion is involved in generating diverse antibody repertoires. And we also, from work from the Lombardo's lab at Columbia University, it's been proposed that enhancer promoter interaction are actually key to regulate olfactory receptor promoter choice. So I think that the concept that enhancers and promoters need to come together in a stochastic manner to do something to that promoter is actually, goes beyond the protocadherin gene cluster, but can actually has implication that really look at how cells utilize this mechanism to generate stochastic expression profiles. So with that, I'd like to thank you for your attention, first of all, and then thank my lab. This was really a team effort of everybody, but particular thank goes to 3 people: a postdoctoral fellow, wonderful postdoctoral fellow in the lab, Leah Kiefer, and 2 fearless technicians who joined the lab at the beginning, Sandy and Gabby. And with that, I'd like to thank you and take any question. Thank you.
Thank you, Daniele. That was fabulous. Questions for Daniele? As people are considering, I'm going to ask a question, Daniele. I'm really intrigued by the model that cohesin using ATP is not, I think if what you're suggesting is not just doing loop extrusion, but it's doing other kinds of work to get out of this repressive environment. Can you speculate on what this other kinds of work might be, if I'm getting this right?
Yeah, absolutely. So I think we have 2 models here. One, it either acts as a cargo, where basically brings perhaps histone demethylases or TET enzymes with it, and so in the way in which it translocates through the locus, then demethylates stochastically 1 or 2 promoters, or it acts to bring together the enhancer and the promoter, where the enhancer actually is a hub for these enzymatic activities. And so these 2 different models we haven't yet fully tested yet, but you could think that yes, you basically uses the energy of ATP to bring together regulatory sequences, either by carrying it with you or by just allowing them to bump into each other more frequently.
So could you bring back Stavros's model of the enhanceosome here? Would you imagine that is actually helping move the whole enhanceosome with it?
Yeah, so of course, in the case of olfactory receptor choice, the situation is even a little bit more complex, because there, these enhanceosomes are generated by bringing together regulatory sequences that are on different chromosomes. And so it turns out that CTCF and cohesin are not needed for that choice, because CTCF and cohesin act in cis. And so it's a different type of mechanism of genome organization that needs to actually cope with the challenge of having to go in trans. But for the BDJ instead, where the actual mechanism occurs in cis, that's in that case, loop extrusion is being the model proposed for actually mediating enhancer promoter interactions and BDJ recombination.
Thank you. Vijay has a question. Vijay, do you want to ask it?
Yeah, okay. Yeah, so Daniele, you guys had this really beautiful experiment where you include CTCF sites, and of course it makes a lot of sense, because when we think about barriers to loop extrusion, CTCF often comes up, but there are actually many other factors that can empirically serve as barriers to loop extrusion, just even by thinking about insulating factors and high Cmaps, right? And so have you guys thought about potentially ectopically introducing other sequences like TF3C binding elements and seeing if those can quantitatively in a different way affect promoter choice, right? Could you effectively tune the insulation strength to tune promoter choice in a quantitative manner?
No, that's a great question. It's a great suggestion. We had not thought about it, but we have thought about using this cluster as a sort of basically framework to study DNA loop extrusion. And a lot of this is actually in collaboration also with the NORA lab here at UCSF, where we could indeed use this as an elegant system to then study the process of DNA loop extrusion and perhaps engineer experiments like the one you're suggesting by tuning the ability of insulation of different factors to see how that then scales to promoter choice. So this is a great suggestion. Thank you, Vijay.
Thank you. And thank you again, Daniele, for a fabulous talk. We'll now move on to our next and final speaker for today, Tatiana Kutateladze from, she's speaking to us from Denver, where the time currently is 12:37 p.m. Welcome, Tatiana.
Thank you. Can you see my slides?
Yes.
Okay, let me just go into presentation mode. All right. Yeah, well, I would like to thank Gita and Danica for inviting me, and I would like, I really enjoyed today's, all talks are fabulous, and look forward to talks tomorrow. And I would like to tell you what we are doing with histone readers, and I will skip a long intro for this meeting. I would like just to mention that my lab, we are interested in the structure function activities of readers, and we know that readers are proteins or protein modules that specifically, or at least selectively, recognize post-translational modifications, and perhaps the most well-studied readers are bromodomains and chromodomains, and we became interested in plant homeodomain or PhD fingers quite a long time ago. In fact, 4 labs simultaneously, including David Alice, Shaw Patel, Orgazani, and my lab found that PhD fingers are capable of binding to methylated histone tails. And the list of epigenetic readers is growing very fast. We know now the whole bunch of those domains that bind to Ptms. But going back to 2006, when together with OR, we found that PhD finger of the inhibitor of growth 2, it's a tumor suppressor, binds to trimethylated lysine 4 of histone H3. This is the crystal structure of the complex. And you could see that trimethyl ammonium group of lysine 4 is caged in the aromatic cage, and this interaction stabilizes the CN3A H-dog complex at promoters of the proliferative genes. And for many years, we studied how single readers interact with single Ptms. But of course, we know that many nuclear proteins contain not one reader, but several in all kinds of combinations. And one of the examples is this MLL4 protein. It's a methyltransferase. It has this C-terminal set domain, and it also has 7 PhD fingers. So the question is, why on earth nature created this protein to have so many PhD fingers. Although we do know that they are not redundant, their functions are not redundant. And let me show you what we know about this 6-1 PhD 6. It does bind to acetylated lysine 16 of histone H3. Oh, sorry, H4. And this is NMR structure of the complex. You could see that acetylated lysine is caged in the hydrophobic groove. These two leucine residues, one tryptophan residue. And we investigate the functional importance of this interaction in collaboration with Kai Gao. What Kai did, they used cells derived from knockout, it's MOF knockout mice. We know that MOF is acetyl transferase that catalyzes, that acetylates lysine 16 of histone H4. And Kai found that 1/3 of MLL4 chromatin binding sites overlap with those chromatin regions enriched in this mark. And another really cool experiment, what Kai did, they again used MOF knockout cells, so there is little acetylation of K16, and then they reintroduced either wild type MOF or catalytically inactive MOF. And we focused on occupancy of MLL4 at the promoters of its target genes. And so what we see again and again for a subset of genes so here is the occupancy of MLL4 in the control cells, and then MOF knockout cells. So we don't see colocalization of MLL4 at this site. Reintroduction of wild type MOF restored localization of MLL4, but not when catalytically dead MOF was added back. And this trend correlates very well with the level of acetylation of K16. We could see pretty good level of this mark in the control cells, substantial decrease in MOF knockout cells, and this level was restored when again wild type MOF was reintroduced, but not when catalytically inactive mutant was reintroduced. And another interesting example of regulation of the catalytic activity by readers we see in P300. P300 is a very well-studied histone acetyl transferase. Acetylates, as far as histones is concerned, although it acetylates not only histone, but non-histone proteins as well. But it primarily acetylates lysine 27 and lysine 18 of histone H3. And we didn't know why. P300 has an interesting combination of readers and other domains. It's so-called a core of this catalytic core of P300. Here is acetyl lysine binding bromo domain, ring finger, PhD finger. Here is the HAT domain, the catalytic domain, and the ZZ domain of unknown function. We collaborate with Xiaobin Shi on this and many other projects, and Xiaobin found that ZZ domains bind to histone H3 tail, and no surprises here. P300 ZZ domain also binds to to unmodified histone H3. It doesn't care whether lysine 4 is methylated or acetylated. The binding is pretty good. We saw the crystal structure of the complex of this ZZ domain bound to the histone H3 tail in orange. And the take-home message of this study is that when ZZ domain binds to the N-terminus of histone H3 tail, alanine 1, arginine 2, T3, blah, 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 it presents the rest of the histone tail in cis to the hot domain. And the distance between the active site of the hot domain and the binding site of ZZ domain is such that only distal lysine residues of histone H3, lysine 27, lysine 18 could reach this active site of HAT. And this distance is way too long for K14, K9, K4 to be primed right here at the catalytic, at the active site of the hot domain. And we also found that both acetyl lysine binding activity of their bromo domain and histone binding activity of ZZ domain are necessary for P300 to associate with chromatin. These very elegant experiments and really solid experiments came from Xiaobin. Here we investigated hot activity of P300, and indeed we confirmed that wild type P300 acetylates lysine 27, lysine 18, those black bars, they correspond to the hot activity of wild type protein. it can also acetylate other lysine residues. We tested just K4, K9, but non-specifically. You could see the difference in fluorescence signal. Here we're talking about units 12, and here it's 0.08. But when we mutated ZZ domain or removed, deleted this domain, and those are red and orange bars, we lost this selectivity toward K27 and K18, but acetylation of, well, non-specific acetylation just continues. When we mutated bromo domain, we see a decrease in HAT activity everywhere. And when we disrupted both ZZ domain and bromo domain, then we see that HAT domain, the HAT activity was pretty much gone. So here is our current model for P300, for the regulation of P300 by the readers. We know that this acetyl lysine binding activity of bromo domain is necessary for the overall HAT function of this protein, but binding of the ZZ domain to the histone H3 tail promotes selective acetylation at lysine 27, lysine 18. We haven't tested lysine 23. We also found that P300 forms condensates in vitro and in vivo. You could see those yellow speckles. They're quite rounded, and FRAP assay confirmed that these speckles have liquid gel-like properties. Those speckles also co-localize with this marked trimethylated lysine 27 of histone H3 to a better degree than it co-localizes with other marks. And importantly, what we found that the catalytic activity of P300 in those vesicles is decreased. And that was totally surprising to us. And as far as what is the mechanism for the formation of these vesicles, we studied this, and we came up with at least two. And both mechanisms involve the catalytic core, which is structured as opposed to intrinsically disordered regions. But in both cases, this auto inhibition loop in the hot domain has to be deacetylated, because acetylation of this loop disrupts formation of those condensates. And now we know that this ability to phase separate formation of vesicles is important for functions of many proteins, including nuclear proteins, chromatin associated proteins. So we follow what the work of Gita in this respect, and other labs, like Rosen. And so here in this study, we focused on those leukemogenic translocations of NUP98 with other partners, and those partners are PHF23, KDM5A, that's what Daniele is interested in, BPTF. So we propose that this ability to form those speckles, vesicles, concentrates those translocation, well, those fusion proteins, and this mechanism could be a reason for overactivation and overexpression of oncogenes. So what happens that this part, PhD finger, of course we know that, well, in this case, all three proteins, their PhD fingers bind quite well to trimethylated lysine 4 histone H3, and we know that this mark is a mark of active gene transcription. But the NUP part, NUP98, forms oligomers, could be oligomerized, and could be important for phase separation. And another example of the importance of reader for leukemogenic mechanisms, we saw it in the case of AF10. And this work, oh, I forgot to mention, I apologize, that in the previous slide, that was work together with two groups, Peter Upland and Greg Wang. And here we worked with Ani Deshpande. We were interested in this part of AF10. It's so-called PZP. It's a reader. It's a combination of PhD1, zinc knuckle PhD2. This part binds to histone H3 tail, and the mechanism is quite unusual because not only residues alanine 1, arginine 2, but a really great stretch of H3 tail up to lysine 27 are involved in the contact with the PZP. So I overlaid here structure from Orgazani lab and my lab, and it's almost seems like this PZP domain almost wrapped well, histone H3 tail almost wraps around this reader. And we know that AF10 translocates with many partners. Ani investigated this translocation with CALM. And what Ani thinks, and what we see again and again, that AF10 can translocate with many partners, but in all cases, this PZP domain is compromised, especially PhD1. There is no single leukemia-associated translocation where PZP is intact. So what Annie did, they reincorporated intact PZP into this chimera, and they saw really surprisingly remarkable, spectacular results when they used, well, they injected mice with cells that were transduced with constructs, whether that have either intact PZP or natural chimera, leukemogenic chimera. And green line corresponds to the construct that has intact PZP. Those animals were not sick, they did not develop leukemia, and they were totally fine. But those animals that did not have intact PZP, they developed leukemia and died soon after the experiments. We also see, not surprisingly, a huge difference in expression level for Hox and Mace genes. And we came up with a model to explain all this. So CALM is known to be targeted and anchored to the promoters of Hox genes through this yet another protein, so-called CRM1. And F10 portion obviously interacts with .1L, that's the methyltransferase, methylates lysine 79 of histone H3. And what was shown by other labs and by Annie's group, that there is an elevated level of this mark, methylated lysine 79, at Hox genes, and that's exactly what we see, and upregulation of Hox genes, and there is no spreading of this complex onto other chromatin regions, so there is overall global reduction of dimethylated lysine 79. However, when we incorporated PZP, now it counterbalances the association, this association, and now we see spreading of this mark onto other daisy. And I will probably skip this a couple of slides. Just wanted to show that we continue working with Jacques Catter on the interplay between histone readers in the Moss-Morph complex. Recently we found that actually Moss-Morph complexes, they acetylate lysine 23, and that was really unexpected. Fabulous experiments came from Jacques' lab that we know quite a bit about acetylation, deacetylation, lysine 4, lysine 9, lysine 14, lysine 27, certainly, but lysine 23 was a little bit not as known. Now we know that Moss-Morph complexes select this mark for acetylation, and I would like to acknowledge my very talented group, current members, former members, our collaborators, without whom this would be absolutely not possible. My R1s from the NIH, and again, thank you for listening. Yeah.
Thank you, Tatiana. There was a ton of exciting data. We already have one question from Danica. Go ahead.
Tanya, beautiful talk. So with leukemogenic fusion, if I got it correctly, the wild type protein differs from the translocated fusion protein by the absence of PHD1 in PZP domain. Is that correct?
Let's say you're talking about AF10?
Yes, AF10.
Yeah, so PHD1 is completely, yeah, there is no translocations that have intact PHD1. Still, zinc finger and PHD2 sometimes they are intact. But you probably know that PhD1 is very important for binding to alanine 1, arginine 2. So that's probably the case.
Thank you.
Thank you, Tatiana. There's a question from the chat, and then we'll go to Phil. The question from the chat is from Anirban Dasgupta. Anirban says, very cool story. I'm a graduate student from India working with one of your former postdocs, Siddharth. Very happy to hear you talking about histone readers. I was wondering if, one, ring domain in tandem with the PhD finger contributes to P300 intra-domain collaborations apart from inhibiting the P300 hat activity. That's question number one. Do you want to answer that first? And then I'll tell you his second question.
Right. So you are working with Siddhartha Roy, with Sid? That is totally exciting. For your question about P300, although we know that this PhD finger does not interact with histones, many labs tried, but yeah, and ring finger based on the structure of that came from Daniele Payne lab, a couple of crystal structures. We do know that ring finger and perhaps PhD, well, at least ring finger is involved in the regulation. It's just the conformational changes.
And his second question is also, do you think the different PhD fingers of MLL4 have different affinities for their histone substrates?
They bind not only to histones, and that's what we are working right now. Yeah, I can tell you for sure that their functions are not redundant.
Phil, do you want to ask your question?
Oh yeah, thank you. Very nice talk, Tatiana. Long time no see. I wanted to kind of follow up on the intriguing idea that the ZZ domain, we used to call it as part of the CH3 region of P300, is sort of a recruitment domain for the H-tail. There's, I guess, another body of work that suggests that that region of P300 is a sort of general transcription factor binding region for things like P53. Do you think that those can bind compatibly? Because I know there's ZZ, and I actually don't remember all the nomenclature. There's something called TAZ2, I think. But is there a way to simultaneously bind the histone tail and a transcription factor? Or if not, could you imagine a scenario where one tail is binding the transcription factor and the other, one ZZ is binding the transcription factor in a nucleosome, and another one is binding, in the P300. So just curious to hear how you would put all that together.
Yeah, TAZ2 is a separate domain. There is a linker between ZZ and TAZ2, and so yeah, there is a good possibility that those two bind to different ligands and enhance, association with chromatin and induce, well, or at least promote transcription. Another curious thing, what we found that the ZZ domain in general, the mechanism of interaction of ligands with ZZ is, the ZZ domain has a very negatively charged groove, and not only histone tail may bind to this groove. So when you brought the issue of P63, I need to go back and
actually it was P53, but yeah, what I can't remember is
P53
which domain, or which because I used to think of them as sort of part of one larger domain called the CH3 domain, but I guess as you
Yeah,
correctly
they are
pointed out,
different. Yeah.
they're now somewhat
Yeah.
viewed as separate. But is it known by you or others whether you can bind two things, like you can bind concurrently to both of them, both of those subdomains within the CH3?
You know, we haven't investigated. Jabin asked us to do something with TAS2, but we haven't even started.
Thank you.
Great. We will end with one last question from Kwang-Woon Lee. Kwang-Woon says, I'm curious if the zinc ions in the ZZ domain affect the peak intensities of the isotopically labeled histone tails when bound.
Well, what I can tell you that zinc ion is absolutely necessary for the structure of this domain. You remove the zinc ion with chelators, the structure will be gone.
Great. So with that, I want to thank Tatiana for a fabulous talk and all the speakers today for taking the time, and I know everybody's very busy, so for taking the time to share with all of us your latest exciting results. Thank you all for participating, and thank you to the audience for participating to their many questions. See you all tomorrow. Take care. Bye-bye.